Dawn filters into the shallows as a diluted blue glow, spreading across a seagrass meadow where currents move with the slow, tidal breathing of the coast. The sand below is pale and fine, interrupted by scattered shells and the faint tracks of nocturnal crabs that have already retreated underground. A few meters above the seabed, suspended in a posture that appears almost casual, a cuttlefish hovers with effortless control. Its oval mantle undulates in subtle ripples, driven by a skirt-like fin that runs along both sides of its body, allowing it to move forward, backward, or remain suspended without the abrupt thrust of jet propulsion. The animal's arms are gathered loosely beneath its head, the two longer feeding tentacles folded invisibly within specialized pockets, coiled and primed. From a distance, it looks like a pale fragment of drifting debris. Only when the light shifts does the illusion fracture, revealing that the surface of its skin is not static camouflage, but a living display panel capable of instantaneous transformation. Beneath the thin, translucent skin of the cuttlefish lies a layered system of cells that function with an efficiency and complexity that rivals engineered display technology. The most visible components are chromatophores, microscopic pigment sacs containing red, yellow, and brown granules. Each chromatophore is encircled by radial muscles controlled directly by motor neurons. When those muscles contract, the pigment sac expands like a tiny umbrella, exposing color to the outside world. When the muscles relax, the sac shrinks and the pigment disappears from view. Tens of thousands of these units cover the animal's body, and they can be activated in coordinated patterns within fractions of a second. Beneath the chromatophores lie iridophores and leucophores, reflective structures that manipulate incoming light. Iridophores use layered plates of protein to create iridescent blues and greens through constructive interference, while leucophores scatter ambient light, acting as a white base layer that enhances contrast. Together, these three tiers allow the cuttlefish not only to alter color, but to adjust brightness, contrast, and even the apparent texture of its skin. As the morning brightens, a small school of juvenile fish drifts into the meadow, picking at plankton and grazing on microscopic growth along the blades of seagrass. The cuttlefish rotates slightly, aligning its body with the orientation of the plants. In an instant, faint bands ripple across its mantle, mimicking the vertical lines of grass stems. Tiny papillae, muscular hydrostatic structures embedded in the skin, rise into low bumps and ridges, breaking the smooth outline of the animal and giving it the appearance of a frayed leaf. This is not passive blending, it is an active reconstruction of the surrounding geometry. The cuttlefish is reading the scene through large W-shaped pupils that control light entry with extraordinary precision. Unlike most vertebrates, its retina does not distinguish color in the conventional sense. Instead, it likely interprets chromatic information indirectly through chromatic aberration, adjusting focus to infer wavelength differences. The result is a paradoxical creature that may be effectively colorblind in the human sense, yet masters of color manipulation in practice. One fish separates from the group, drawn toward a patch of disturbed sand. The cuttlefish shifts into what researchers describe as a dynamic display. Waves of contrast sweep along its body in rapid succession, a phenomenon sometimes referred to as passing cloud. Dark and light bands pulse forward, creating the illusion of motion, even though the animal itself remains almost perfectly still. To the fish, whose visual system is tuned to detect movement as a cue for both prey and predator, this controlled flicker becomes a focal point. The display is not random. It is timed and modulated, the intensity and frequency adjusted to exploit the neural processing of the observer. Many reef fish rely on quick escape responses, mediated by specialized neurons that trigger rapid tail flicks at the first sign of threat. By presenting a moving pattern that does not correspond to actual displacement, the cuttlefish may overload or misdirect those circuits, inducing hesitation. In the open water, hesitation is often fatal. The surface of the cuttlefish now exhibits a more complex transformation. Iridophores begin to reflect polarized light in specific orientations. Polarization refers to the alignment of light waves along particular planes, a property invisible to human eyes but detectable by many marine animals. Cuttlefish possess photoreceptors sensitive to polarization, and they can both perceive and generate polarized patterns. Across the mantle, bands of polarized reflection shift in alternating directions, forming a hidden signal that may be conspicuous to the fish while remaining subtle in overall brightness. These polarized displays can create a shimmering, almost hypnotic effect, not by dazzling in intensity, but by interfering with the prey's ability to interpret edges and motion correctly. The fish drifts closer, its pectoral fins fluttering as it tilts to inspect what appears to be a patch of animated seagrass. In that narrowing distance, the cuttlefish's physiology transitions from deception to attack. The body angle adjusts minutely, aligning the line of fire. Within the arm crown, two specialized tentacles extend slightly forward inside muscular sheaths. These tentacles are distinct from the 8 shorter arms. They are longer, elastic, and terminate in expanded clubs lined with suction cups arranged in precise rows. When deployed, they can double their length in a fraction of a second. The mechanism relies on rapid muscular contraction combined with stored elastic energy in collagen fibers, allowing for explosive extension without sacrificing accuracy. Unlike many predators that must chase, the cuttlefish compresses space into a single, decisive event. The moment arrives without warning. The tentacles launch, propelled by a sudden increase in internal pressure within the mantle cavity, while the fins remain almost motionless. The strike is so fast that the water barely registers disturbance. The clubs clamp onto the fish's body with adhesive suction, and the tentacles retract immediately, drawing the prey into the forest of shorter arms. These arms, each equipped with additional suction cups and sensitive chemoreceptors, secure the fish and maneuver it toward a sharp, chitinous beak hidden at the center of the arm crown. The beak, similar in structure to that of a parrot, shears through scales and flesh. Salivary glands introduce enzymes, and in some species, mild venom that subdues struggling prey. The entire sequence, from the first pulse of chromatic display to the immobilization of the fish, unfolds in less time than it takes for a human observer to inhale. What appears as a simple ambush is, in fact, the culmination of millions of years of evolutionary refinement. The cuttlefish belongs to the class Cephalopoda, a lineage that includes octopuses and squids, known for large brains relative to body size and sophisticated nervous systems. In cuttlefish, the neural circuitry required to control chromatophores is immense. Each chromatophore can be addressed individually or as part of coordinated fields, and the brain integrates visual input with motor output at extraordinary speed. Unlike reptiles or fish that rely on hormonal changes for color shifts that take minutes or hours, cuttlefish operate on the scale of milliseconds. Their skin is effectively an extension of their nervous system, a distributed organ of perception and communication. Yet the chromatic system serves functions beyond hunting. As the captured fish is consumed, another cuttlefish glides into view at the edge of the meadow. Instantly, the first individual alters its display, darkening one side of its body while maintaining a neutral pattern on the other. This phenomenon, sometimes referred to as split signaling, allows the animal to present different messages simultaneously. The side facing the newcomer may exhibit bold, high-contrast patterns associated with aggression or territorial assertion, while the opposite side remains camouflaged against the sand to avoid attracting attention from larger predators such as groupers or rays. In this layered communication, color becomes language, texture becomes punctuation, and polarization adds a hidden channel only certain receivers can decode. The meadow itself is not a static backdrop, but an ecosystem structured by subtle forces. Seagrass stabilizes sediment, providing refuge for juvenile fish and invertebrates. By hunting selectively among these inhabitants, cuttlefish influence population dynamics, potentially affecting grazing pressure on the grass and the distribution of small crustaceans. Their role as mid-level predators connects lower trophic levels to larger consumers. Cuttlefish are prey for dolphins, larger fish, and even seabirds when they venture into shallower water. Their mastery of camouflage is therefore as essential for survival as their hunting precision. When threatened, they can eject a cloud of ink composed of melanin and mucus, creating a decoy shape that lingers while the animal jets away. In some cases, the ink may also interfere chemically with a predator's sense of smell, adding another sensory layer to their defense. As the tide shifts and the sun climbs higher, the meadow becomes brighter, and the cuttlefish's patterns grow more subdued. It settles partially into the sand, lowering its body until only the eyes and the upper mantle remain visible. The chromatophores contract, leaving a pale, speckled pattern that matches the grain of the substrate. Papillae flatten, smoothing the silhouette. From above, even a trained eye would struggle to distinguish animal from sediment. Yet beneath this stillness, neural circuits remain active, sampling light, detecting movement, adjusting polarization. The stealth fighter of the shallows does not rely on speed alone, but on information control. It manipulates what others see, how they interpret it, and how long they hesitate. In this choreography of light and nerve impulses, the cuttlefish reveals a principle that extends beyond a single species. Survival in the marine environment often depends not merely on strength or endurance, but on the capacity to shape perception itself. By converting skin into a responsive display and integrating polarized light into both signaling and deception, the cuttlefish occupies a unique position among predators. It is at once visible and invisible, aggressive and restrained. A hunter that conquers not through brute force, but through neurological misdirection. In the quiet meadow, where currents carry the scent of plankton and the shadows of passing fish drift across the sand, the cuttlefish hovers as a reminder that the most sophisticated weapons in nature are often those that operate at the level of sensation, bending light and reflex into tools of survival. The reef at mid-morning is not quiet, but it is orderly. Sunlight penetrates the surface in fractured shafts, scattering through suspended plankton and illuminating a mosaic of coral heads, sponge towers, and sand channels that form the architecture of a living city. Reef fish patrol their territories with predictable routes. Grazing parrotfish grind coral into sand with audible crunches, and cleaner wrasses hover near their stations, advertising hygiene services to passing clients. Beneath this visible choreography, however, a smaller drama unfolds within crevices that most eyes pass over. In the shadowed entrance of a narrow burrow, excavated beneath a slab of coral rubble, a mantis shrimp waits, its body coiled within a tunnel just wide enough to conceal its segmented armor. Only its eyes protrude beyond the threshold, mounted on mobile stalks that pivot independently and scan the reef with unsettling precision. The mantis shrimp is a crustacean, not a true shrimp, but a stomatopod, a lineage that diverged from other crustaceans hundreds of millions of years ago. Its body is armored in overlapping plates that offer protection without sacrificing mobility, and its thoracic appendages are folded tightly beneath its head, locked in a configuration that resembles a cocked mechanism rather than a resting limb. Yet the most arresting feature remains its eyes. Each eye sits atop a muscular stalk capable of rotating in multiple planes, granting a field of view that approaches full coverage around the animal's body. Unlike vertebrate eyes, which process images with a layered retina connected to a single optic nerve bundle, mantis shrimp eyes are structured into three distinct regions, separated by a horizontal band of specialized ommatidia that function almost like a scanning strip. This midband allows the eye to sample spectral and polarization information with extraordinary resolution. Within those compound eyes are photoreceptor cells tuned to an array of wavelengths that far exceeds the trichromatic system familiar to humans. Where humans rely on three types of cone cells sensitive to red, green, and blue light, mantis shrimp possess 16 distinct photoreceptor classes, each tuned to specific segments of the visible and near ultraviolet spectrum. These receptors do not simply expand the palette of color, they partition light into narrower bands, allowing rapid discrimination without complex neural comparison. Recent research suggests that mantis shrimp may not process color in the same way vertebrates do, comparing signals across cones to create a blended perception. Instead, each receptor type may function more as a labeled channel, enabling the animal to identify wavelengths directly and almost instantaneously. In the shifting light of the reef, where reflections from coral and water distort color cues, this direct detection can provide a decisive advantage. More remarkable still is their ability to perceive circularly polarized light. Many marine animals can detect linear polarization, which arises when light waves align along a particular plane after scattering through water or reflecting off surfaces. Circular polarization, by contrast, involves the rotation of light waves in a helical pattern as they travel, a property rarely generated in nature and almost entirely invisible to vertebrate vision. Mantis shrimp possess specialized photoreceptors with microvillar structures arranged in a way that allows detection of this helical rotation. Some species even produce circularly polarized signals from reflective structures on their bodies, effectively communicating in a visual channel that most predators and competitors cannot perceive. In the densely populated reef, where color signals are abundant and easily intercepted, circular polarization becomes a private frequency, an encrypted visual language. From its burrow entrance, the mantis shrimp surveys the corridor of sand that runs between coral heads. A small crab emerges from beneath a rock, pausing to probe the substrate with its antennae. The crab's exoskeleton reflects polarized light differently than the surrounding sand, a subtle cue invisible to most reef fish, but stark to the mantis shrimp's eyes. The stalks swivel independently, triangulating distance and orientation. Each eye alone can gauge depth because the midband samples different angles of incoming light, creating a form of trinocular vision within a single organ. While vertebrates rely on binocular disparity between two eyes for depth perception, mantis shrimp can judge range with one. This capacity allows them to remain partially concealed while maintaining precise spatial awareness. Inside the burrow, the appendages remain folded, but tension accumulates. In species known as smashers, the second thoracic appendages are modified into calcified clubs, thickened with mineralized layers that combine hardness with resilience. These clubs are not swung by simple muscular force. Instead, the mantis shrimp employs a latch and spring mechanism. Large extensor muscles slowly load elastic structures composed of chitin and specialized proteins within the appendage. A locking system holds the appendage in place while potential energy builds. When released, the stored energy converts into kinetic force in an almost instantaneous arc. The strike is so rapid that it surpasses the acceleration of a bullet relative to body size, reaching speeds that generate shockwaves in the surrounding water. As the crab shifts slightly closer to the burrow, perhaps drawn by drifting organic debris, the mantis shrimp's eyes narrow their focus. There is no warning display, no preliminary movement that would alert the prey. The latch releases. The club extends in a blur, traveling through water so quickly that it creates a low-pressure region along its path. In that brief instant, the pressure drops enough to cause cavitation, forming vapor bubbles that collapse violently as pressure equalizes. The implosion of these bubbles generates a secondary shockwave and, under certain conditions, a flash of light known as sonoluminescence. This phenomenon, in which collapsing bubbles emit photons, is rarely visible in the wild but measurable in controlled environments. It represents the conversion of mechanical energy into light, a byproduct of speed so extreme that water itself briefly tears apart. The crab's carapace fractures under the combined force of the direct impact and the cavitation shock. Even if the initial blow does not fully disable the prey, the secondary shockwave can deliver additional trauma, disrupting tissue and neural function. The mantis shrimp retracts its appendage immediately, ready to strike again if necessary. Often, a single hit suffices. The crab collapses, legs folding inward as the mantis shrimp drags it into the burrow. Within the confines of the tunnel, the prey will be dismantled with methodical efficiency, the armored plates cracked open to expose nutrient-rich tissues. This strike is not merely an act of brute force, but the outcome of precise calibration. The mantis shrimp must judge distance accurately to time the release of the latch so that the club meets its target at the optimal angle. Too early or too late, and the blow may glance off, wasting stored energy and exposing the animal to counterattack. The energy required to load the appendage is metabolically costly, and repeated strikes without success can deplete reserves. Thus, the mantis shrimp's visual system and mechanical weapon are tightly integrated components of a single predatory strategy. Vision guides the aim. Stored elastic energy delivers the verdict. Not all mantis shrimp are smashers. Some are spearers, with elongated barbed appendages designed to impale soft-bodied prey such as fish. These species often inhabit sandy burrows in deeper or more open habitats, where ambush tactics rely on concealment and sudden extension rather than blunt force. Yet even among smashers, the reef environment shapes behavior. In territories where coral rubble is abundant, individuals may reinforce burrow entrances with fragments of shell and stone, constructing fortifications that reduce the risk of intrusion by octopuses or larger crustaceans. Territorial disputes between mantis shrimp can escalate into ritualized displays, where individuals raise their appendages and expose reflective patches that may include polarized signals. In some cases, they engage in controlled exchanges known as merrill spreads, assessing each other's size and strength without immediately resorting to destructive strikes. The reef community is aware of the mantis shrimp's presence, even if it cannot fully comprehend the details of its sensory world. Fish often avoid hovering directly in front of burrow openings, and octopuses probing crevices may withdraw abruptly if they encounter the hardened edge of a mantis shrimp's club. At the same time, the mantis shrimp is not invulnerable. Larger fish and cephalopods can overpower it if they breach the burrow, and energy expenditure in maintaining a territory and loading its weapon demands consistent access to prey. Reproductive cycles introduce additional pressures, as females brood eggs within the burrow, fanning them with pleopods to ensure oxygenation and prevent fungal growth. During brooding, feeding opportunities diminish, and the female may fast, relying on stored energy reserves. As afternoon light shifts and shadows lengthen across the reef, the mantis shrimp resumes its vigil. The eyes remain in constant motion, sampling wavelengths and polarization patterns that paint a reality richer and more complex than human perception can reconstruct. To the mantis shrimp, the reef is not only a tapestry of color, but a matrix of hidden signals, a field of encoded reflections and rotational light cues that reveal the texture and orientation of surfaces. In this environment, the ability to detect circular polarization may assist in recognizing conspecifics, distinguishing prey against complex backgrounds, or interpreting subtle changes in water column properties. The strike, though spectacular, is only the final expression of a deeper adaptation, a sensory system attuned to nuances of light that most animals never experience. The supersonic sniper of the reef does not chase its prey across open water, nor dominate through size. It relies instead on patience, perception, and the controlled release of stored energy. In the narrow space between coral and sand, where centimeters determine success or failure, the mantis shrimp has refined a strategy that compresses physics into a single motion. Light bends through water, pressure waves ripple outward, and for a fleeting fraction of a second, sound becomes light in the collapse of cavitation bubbles. In that flash, invisible to most but measurable in principle, the reef reveals the extremes to which evolution will push a body, when survival depends on speed beyond sight. The northern summer light stretches across the subarctic sea in long silver bands, illuminating a surface that appears calm until the first exhalation breaks it. A column of mist erupts into the air, hangs for a breath, and dissolves. Beneath that plume, a humpback whale drifts in near vertical suspension, its immense pectoral fins angled slightly outward like stabilizers on a slow-moving aircraft. The water here is rich with plankton, fertilized by upwelling currents that carry nutrients from deep basins toward the light. That productivity has triggered a chain reaction. Swarms of small schooling fish, often herring or sand lance, compress into dense shimmering clouds as they feed. Above them circle gulls and shearwaters, watching for a disturbance below. And below them, invisible in the green water column, several humpback whales position themselves with the precision of engineers preparing a construction project. The humpback whale is not merely a predator in this setting. It is an architect of temporary structures built from air and sound. Each whale in this loose coalition weighs as much as a small building and must consume enormous quantities of prey during the feeding season to build the fat reserves that will sustain migration and reproduction later in the year. Unlike toothed whales that hunt individual prey with echolocation clicks, humpbacks are baleen whales. Instead of teeth, they possess plates of keratin that hang from the upper jaw like dense curtains. These baleen plates act as a sieve. When a whale engulfs a volume of water filled with fish, It closes its mouth and forces the water out through the baleen, trapping prey inside. The efficiency of this system depends on concentrating prey into tight masses before the engulfment. A dispersed school is wasteful. A compressed sphere is a banquet. Several whales descend beneath the fish, their massive bodies tilting downward in a slow spiral. The group does not move randomly. They adjust spacing and depth in relation to one another, maintaining a loose circular formation. From each blowhole, bursts of air stream downward into the water, released not as a single chaotic plume, but in controlled pulses. These bubbles do not rise immediately in a straight column. Instead, the whales angle their bodies and swim in tightening circles as they exhale, producing a rising cylinder of bubbles that expands and then narrows, forming a luminous barrier around the fish. To a human observer from above, the surface may show only faint turbulence. Underwater, the effect is unmistakable, a vertical wall of shimmering spheres that distort light and create a boundary the fish hesitate to cross. Fish are sensitive to abrupt visual contrasts and to pressure changes. A curtain of bubbles presents both. The bubbles refract and scatter light, creating a chaotic visual field that confuses schooling cohesion. At the same time, the ascending column produces microcurrents and vibrations that the fish detect through their lateral line system, a sensory organ attuned to water displacement. When confronted with this barrier, many fish respond by tightening their formation and moving inward, away from the perceived threat. The whales exploit this reflex. By coordinating the radius and speed of their spiral ascent, they determine the diameter of the bubble net. Too wide, and the fish remain loosely packed. Too tight, and the fish may panic and break through gaps. The geometry must match the size and density of the prey school. In that sense, bubble net feeding is a form of acoustic and spatial calculation, an embodied geometry executed in 3 dimensions. Within the growing cylinder, one whale emits a series of low-frequency vocalizations. These calls are not the long, complex songs associated with breeding grounds, but shorter feeding calls, structured sounds that propagate through the water column. Sound travels faster and farther in water than in air, and humpback whales are exquisitely adapted to both produce and perceive it. Their laryngeal sac and associated structures allow them to generate powerful tones that can influence prey behavior. Some studies suggest that specific feeding calls may drive fish downward or inward, reinforcing the compression created by the bubble wall. The timing of these calls relative to bubble release is not arbitrary. It is synchronized. One whale may function as a signal leader, initiating the ascent once the net has reached sufficient density. The coordination required is extraordinary. These whales are not bound by visible ropes or mechanical constraints. They rely on shared cues. The feel of water pressure changes generated by nearby bodies, the sound of exhaled bubbles, the acoustic signals exchanged through the water, and perhaps subtle visual glimpses through the refracted curtain. Each whale must maintain awareness of the other's positions while adjusting its own trajectory. The circle tightens. The bubble wall thickens. Inside, the fish mass becomes a pulsing orb, compressed from all sides. Gulls begin to scream overhead, sensing the impending surge. Then, almost simultaneously, the whales accelerate upward from below the trapped fish. Their mouths open to nearly right angles, lower jaws expanding as ventral pleats along the throat stretch like accordion folds. This expansion allows the oral cavity to balloon outward, engulfing a volume of water and prey that can exceed the whale's own body mass. The ascent is powerful but controlled. If a whale misjudges its timing, it risks colliding with a partner or disrupting the net prematurely. Instead, their trajectories converge just beneath the surface, where the fish are most tightly packed. The surface explodes into white water as multiple whales breach through the bubble curtain, mouths agape, engulfing the concentrated school in a single coordinated lunge. As they close their jaws and roll slightly to the side, water gushes outward through the baleen plates. Fish, now unable to escape, remain trapped inside the cavernous mouth. The whales swallow, reset, and often dive again to repeat the process. Each cycle refines the geometry. If the prey density decreases, the whales may adjust the diameter of the next bubble net or alter the depth at which they initiate the spiral. This is not rigid instinct executed identically each time. It is adaptive behavior shaped by experience and social learning. Calves observe their mothers participating in these coordinated hunts, absorbing the choreography over successive seasons. In some regions, bubble net feeding is common. In others, humpbacks employ entirely different techniques, suggesting cultural variation within the species. The engineering analogy is not metaphorical exaggeration. By releasing air in a calculated spiral while swimming in a defined path, the whales construct a dynamic structure with measurable physical properties. The bubble net has density gradients, flow patterns, and optical effects that influence prey behavior. The whales must solve a three-dimensional problem: how to contain a moving target that responds to pressure, light, and sound. Their solution integrates biomechanics, acoustics, and hydrodynamics. The exhaled air originates from lungs adapted for deep diving, capable of collapsing under pressure to avoid nitrogen absorption that would otherwise cause decompression sickness. When released at depth, the air expands as it rises, contributing to the vertical continuity of the net. The whales intuitively account for this expansion by adjusting the depth and speed of exhalation. Above the surface, the spectacle lasts seconds. Below, it is the culmination of minutes of positioning and signaling. The fish that escape the first lunge often regroup, only to find themselves encircled again. For the whales, efficiency matters. During feeding season, they may consume vast quantities of fish each day, converting that protein and fat into the thick blubber layer that insulates them during long migrations to tropical breeding grounds. In warmer waters, humpbacks rarely feed. They rely on stored energy accumulated during these high-latitude months. Thus, bubble net feeding is not merely a clever tactic. It is a critical strategy that underpins the species' life history, linking polar productivity to equatorial reproduction. The social dimension adds another layer. Not all humpbacks participate equally in every net. Some individuals specialize in producing the bubble wall, others in vocal signaling, others in lunging from specific angles. Research using tagging technology has revealed consistent roles among certain groups, suggesting that cooperative feeding may involve learned positions within a team. These associations can persist across seasons. While humpbacks are not considered permanently social in the same way as some toothed whales, these temporary alliances demonstrate a level of coordination that approaches what might be described as collective intelligence. Each whale contributes to a structure that none could build alone with the same efficiency. As the sun lowers toward the horizon, the sea calms again. The gulls disperse and the fish schools thin, their numbers reduced. One whale lingers near the surface, rolling slowly as if surveying the aftermath. Beneath the placid water, faint remnants of bubbles drift upward and vanish. The architecture dissolves as quickly as it formed, leaving no trace except in the altered density of fish and the satiation of massive bodies that will soon begin their long journey south. In this ephemeral construction of air and sound, the humpback whale reveals a strategy that transcends brute force. It is a builder of invisible walls, a mathematician of motion and pressure, shaping water itself into a tool. Through synchronized breath and shared timing, these marine mammals transform a diffuse swarm into a contained sphere, demonstrating that in the open ocean, intelligence often manifests not in solitary cunning, but in the harmonized geometry of a group working as one. Along the temperate southern coastline, where cool currents sweep across limestone reefs and forests of kelp rise toward filtered sunlight, the water moves with a deliberate rhythm. Long fronds of brown algae bend and recover in slow arcs, tracing invisible patterns dictated by swell, tide, and wind. This is not the riotous brightness of a tropical coral reef, but a subtler landscape of olive greens, amber light, and drifting particulate. The water carries a faint haze, softening outlines and blending forms into one another. Within this suspended world, visibility depends not on brilliance, but on motion. What moves abruptly is noticed. What moves in harmony with the background is absorbed into it. Here, survival is less about speed and more about synchronization. Suspended among the kelp blades, is a creature that appears less like an animal and more like an accident of growth. The leafy seadragon drifts upright, its elongated snout angled slightly downward, its body adorned with elaborate appendages that resemble torn fragments of seaweed. Each projection extends from the body in thin, flattened lobes, branching in irregular patterns that mimic the serrated edges of surrounding algae. These appendages are not fins in the conventional sense. They do not propel the animal. They are extensions of skin and connective tissue, richly vascularized but structurally delicate. Their purpose is visual deception. At a glance, the seadragon is indistinguishable from the detritus of the kelp forest. Unlike many fish that rely on rapid escape or armored defenses, the leafy seadragon embodies passive mimicry. It does not hide behind objects. It becomes the object. Its body coloration mirrors the gradients of the habitat, with mottled yellows, greens, and faint purples echoing the hues of sunlit and shaded algae. Subtle striping along the torso breaks up the continuity of its outline, aligning with the linear patterns of kelp stems. Even the eye, often a conspicuous feature in prey species, is ringed with small protrusions that disrupt its circular shape. In a habitat where predators include larger fish and cephalopods equipped with keen vision, eliminating recognizable edges is critical. Movement is the most revealing signal in aquatic environments. Predators often detect prey by isolating motion that contrasts with the background flow. The leafy seadragon addresses this challenge not by remaining rigid, but by matching the frequency and amplitude of its surroundings. Its propulsion comes primarily from a small dorsal fin located near the back of the body and a pair of pectoral fins near the head. These fins beat rapidly, but with such transparency that they are nearly invisible in the water. The rest of the body remains largely still, drifting with currents. When a swell passes through the kelp forest, the seadragon tilts and sways in synchrony, its appendages lagging slightly behind the central body in a manner consistent with flexible plant tissue. This slight delay, a product of water resistance acting on the thin lobes, creates a convincing illusion of botanical origin. The precision of this mimicry suggests evolutionary fine-tuning beyond superficial resemblance. In the kelp forest, oscillation frequencies vary depending on depth and proximity to open water. Near the surface, wave energy produces faster, more chaotic movement. Deeper in the forest, motion is slower and more uniform. Leafy seadragons tend to inhabit zones where currents generate steady, rhythmic swaying rather than violent turbulence. Their morphology appears optimized for these conditions. The length and flexibility of the appendages, combined with the buoyancy control afforded by a swim bladder, allow them to hover at specific depths with minimal muscular effort. By adjusting gas volume within the bladder, they achieve near neutral buoyancy, reducing the need for active swimming and minimizing disruptive motion. The concept of passive mimicry extends beyond visual camouflage. Many marine predators rely on lateral line systems to detect water displacement caused by moving prey. A fish that darts or pulses generates distinct hydrodynamic signatures. The leafy seadragon's strategy of drifting with ambient flow reduces relative motion between its body and the surrounding water. Instead of cutting through currents, it allows currents to carry it. When it does move intentionally, the motion is incremental and aligned with prevailing flow, preventing the creation of abrupt pressure waves. In this way, the animal avoids triggering what might be described as biological radar, the multisensory detection systems of its predators. Yet the seadragon is not solely a passive participant in this environment. It must feed. Its diet consists primarily of small crustaceans such as mysid shrimp, which hover in loose aggregations among algae. These prey items are alert and capable of rapid escape. The seadragon approaches them with slow, deliberate adjustments, maintaining the same oscillatory pattern as nearby fronds. Its elongated snout ends in a small, toothless mouth, adapted for suction feeding. When within range, it expands the buccal cavity rapidly, creating negative pressure that draws the shrimp inward. The strike is subtle, lacking the explosive drama of a mantis shrimp or the engulfing lunge of a whale. It is a quiet intake, a brief interruption in the steady sway. The success of this feeding method depends on remaining undetected until the final moment, reinforcing the value of precise motion matching. Reproduction introduces another layer of complexity to the sea dragon's life. In a reversal of roles common among syngnathiform fishes, which include seahorses and pipefish, the male leafy sea dragon carries the fertilized eggs. During courtship, the pair performs a synchronized dance, aligning their bodies and swaying together in the current. This ritual likely ensures compatibility and timing, as egg transfer requires precise positioning. The female deposits bright pink eggs onto a specialized brood patch located on the underside of the male's tail. This patch becomes vascularized, supplying oxygen and removing waste from the developing embryos. For weeks, the male continues to drift among the kelp, now bearing a cluster of conspicuous eggs that contrast with his camouflage. At first glance, the eggs appear to compromise the strategy of invisibility. Their color stands out against the muted tones of the habitat. Yet the brood patch is positioned on the underside of the tail, often oriented toward the substrate or concealed within denser foliage. The male's continued commitment to motion matching reduces the likelihood of detection. Moreover, the bright coloration may serve a function in intraspecific recognition during the breeding season, signaling reproductive status to potential mates while remaining relatively hidden from predators that view the sea dragon from lateral or dorsal angles. This delicate balance between concealment and communication underscores the evolutionary negotiation between survival and reproduction. The kelp forest itself is a dynamic ecosystem shaped by temperature, nutrient availability, and seasonal light cycles. Temperate waters can fluctuate significantly, and thermoregulation in ectothermic fish depends on behavioral adjustments rather than internal heat production. Leafy sea dragons select microhabitats that buffer extreme temperature shifts, often remaining within the structural complexity of the forest, where water movement is moderated. Their metabolic rate aligns with the cooler environment, supporting a lifestyle that favors low energy expenditure over bursts of activity. In warmer periods, when metabolic demands increase, the abundance of small crustaceans may also rise, offsetting the energetic cost. Predation pressure persists despite the effectiveness of mimicry. Larger fish capable of penetrating dense kelp may occasionally detect a sea dragon, particularly juveniles whose appendages are less developed. The success of passive mimicry increases with maturity as the appendages become more elaborate and coloration stabilizes. Juveniles often inhabit slightly different zones, perhaps reflecting a trade-off between growth opportunities and predation risk. This ontogenetic shift highlights that mimicry is not a static trait, but a developmental process refined over time. In moments of stillness, when the current slackens and kelp blades hang vertically, the sea dragon remains suspended, appendages gently undulating in residual flow. Its eye rotates independently, scanning for movement without altering the body's posture. The illusion is so complete that even a trained observer may lose sight of it within seconds. The animal does not flee, does not confront, does not assert dominance. Its strategy is to dissolve into context, to erase the distinction between organism and environment. In a marine world where many species advertise their presence through color, sound, or force, the leafy sea dragon represents an alternative philosophy. It survives by relinquishing individuality to the pattern of the forest. Every ridge, lobe, and oscillation is calibrated to the physics of water and the perceptual biases of predators. By evolving to move with the exact frequency of swaying seaweed, it transforms vulnerability into invisibility. In doing so, it reveals a deeper principle of evolution within complex habitats. Sometimes the most effective defense is not to resist detection, but to align so perfectly with the background that detection never begins. In the shallow estuaries where rivers surrender their sediment to the sea, mangrove forests rise from brackish water like a lattice of living stilts. Their roots arch downward in tangled columns, creating a maze of submerged corridors that trap drifting leaves, crustaceans, and juvenile fish seeking shelter from open water predators. Light penetrates this habitat in broken fragments, filtered through leaves above and refracted by suspended silt below. The water is warm, often carrying the faint brown tint of tannins leached from decaying vegetation. Visibility is limited, yet life thrives in these submerged forests, because structure offers refuge, and refuge offers survival. It is within this intricate labyrinth that one of the ocean's most deceptively simple organisms navigates with astonishing precision. A box jellyfish drifts into view between the vertical pillars of mangrove roots, its transparent bell pulsing rhythmically as it propels itself forward. Unlike the passive drifting of many jellyfish species that rely entirely on currents, this animal swims with intent. The bell contracts and relaxes in steady cycles, expelling water in controlled jets that allow directional movement. From each corner of its cube-shaped body trail, long, delicate tentacles armed with microscopic stinging cells. The animal appears fragile, almost ethereal, yet it is both an efficient predator and one of the most potent venomous organisms in marine ecosystems. More surprising than its venom, however, is its sensory capacity. Despite lacking a centralized brain, the box jellyfish possesses a distributed intelligence that enables it to avoid obstacles, hunt actively, and maintain orientation within a visually complex environment. At each of the four corners of the bell hang sensory structures known as rhopalia. These are not simple patches of nerve tissue, but compact processing hubs that integrate information from multiple sensory organs. Each rhopalium contains 6 eyes, resulting in a total of 24 eyes distributed symmetrically around the body. Some of these eyes are simple pit eyes that detect light intensity, while others are lens-bearing camera-type eyes capable of forming images. 2 of the eyes in each rhopalium include corneas, lenses, and retinas, with photoreceptors sensitive to different wavelengths, allowing for a form of color discrimination. This capacity challenges long-held assumptions that complex vision requires a centralized brain. Instead, the box jellyfish distributes processing across its 4 hubs, each acting as a localized center of interpretation and response. As the jellyfish approaches a dense cluster of mangrove roots, the water ahead darkens with shadow. The upper lens eyes, oriented upward, detect the contrast between the bright canopy filtered through leaves and the darker void beneath the roots. The lower lens eyes scan horizontally, resolving vertical silhouettes against the lighter background. When a root looms too close, the jellyfish adjusts its trajectory, altering the angle of its next contraction to veer away. These corrections are subtle and continuous. The animal does not collide randomly and bounce off surfaces. It anticipates structure. Experiments in controlled environments have demonstrated that box jellyfish can learn to associate certain visual patterns with obstacles, reducing collision frequency over time. Such learning, occurring without a brain in the vertebrate sense, implies that neural circuits within each rhopalium can modify responses based on experience. The nervous system of the box jellyfish forms a ring around the bell, connecting the rhopalia in a circular network. This nerve ring coordinates muscle contractions and integrates sensory input from the tentacles as well as the eyes. While there is no centralized processing organ equivalent to a vertebrate brain, the nerve ring and rhopalia together create a decentralized architecture capable of complex behavior. Signals from one rhopalium can influence the contraction pattern across the bell, ensuring coordinated swimming. In effect, intelligence is not concentrated in a single node, but distributed across multiple hubs, each contributing partial analysis that converges through shared pathways. Within the mangrove habitat, this distributed system proves advantageous. The roots create a 3-dimensional maze with irregular spacing, and currents shift unpredictably with tides. A centrally controlled system might struggle with the constant need for rapid, localized adjustments. In contrast, each rhopalium can process visual cues from its own quadrant and trigger immediate motor responses without waiting for a distant command center. The jellyfish becomes a composite of semi-autonomous units working in concert. This architecture resembles decentralized networks in other biological systems where redundancy and parallel processing enhance resilience. As the jellyfish glides past a submerged root, a small fish darts into view, attracted to plankton, illuminated in a shaft of light. The tentacles trailing behind the bell are nearly invisible, thin as threads yet extending several meters in some species. Embedded within them are nematocysts, specialized cells that contain coiled harpoon-like structures loaded with venom. When triggered by mechanical or chemical cues, a nematocyst fires with extraordinary speed, penetrating the prey's tissue and delivering toxins that disrupt ion channels in nerve and muscle cells. The venom of certain box jellyfish species can cause rapid cardiovascular collapse in larger animals, an outcome that underscores the potency of these microscopic weapons. For small fish, paralysis occurs almost instantly. The jellyfish's swimming behavior during hunting differs subtly from its navigation pattern. Instead of weaving tightly through roots, it maintains a straighter path, allowing tentacles to trail widely and increase the chance of contact. The lens eyes detect vertical contrast and movement, guiding the animal toward areas where prey density is higher. When a fish brushes a tentacle, the nematocysts discharge, and the fish convulses briefly before succumbing. The jellyfish senses the disturbance through mechanoreceptors along the tentacles and adjusts its movement to draw the captured prey toward the oral arms at the center of the bell. There is no expression of intent in the human sense, yet the sequence reveals coordination between vision, touch, and motor response. Light conditions within mangrove forests vary dramatically between day and night. During daylight, the canopy creates patchy illumination. At dusk, contrast diminishes and visual cues shift. Box jellyfish exhibit behavioral changes correlated with light intensity. Some species retreat to deeper water at night, while others alter swimming patterns to remain within preferred brightness levels. The upper lens eyes are particularly sensitive to the angle of incoming light, enabling the jellyfish to maintain orientation relative to the surface. In experimental settings, removal of certain eyes impairs the animal's ability to avoid obstacles, demonstrating that each visual organ contributes specific information to the overall navigation strategy. Thermal conditions in estuarine environments can fluctuate with tides and freshwater influx. Although box jellyfish are ectothermic and cannot regulate internal temperature through metabolic heat, they respond behaviorally to thermal gradients. Warmer water often increases metabolic rate, accelerating bell contractions and potentially raising energy demands. In response, jellyfish may seek zones with optimal temperature that balance activity and efficiency. The distributed nervous system likely facilitates rapid local responses to such gradients, as sensory input from one side of the bell can adjust swimming orientation without requiring centralized deliberation. Predation risk also shapes behavior. Juvenile sea turtles and certain fish species feed on jellyfish, though the venom of box jellyfish deters many potential predators. The transparency of the bell provides camouflage in sunlit water, reducing visibility from above. Within the mangrove maze, the vertical roots create visual clutter that further conceals the jellyfish's outline. The lens eyes enable the animal to maintain distance from surfaces where predators might lurk, minimizing entanglement and vulnerability. In this sense, vision serves not only hunting, but also self-preservation. Reproduction adds another dimension to the life cycle. Box jellyfish release gametes into the water column, where fertilization occurs externally. The resulting planular larvae eventually settle onto substrates and transform into polyps, small sessile forms that resemble miniature anemones. These polyps can reproduce asexually, budding to increase numbers before undergoing metamorphosis into free-swimming medusae. The transition from stationary polyp to mobile jellyfish involves dramatic reorganization of tissues and nervous structures. The development of Rhopalia and the nerve ring during this metamorphosis underscores that the distributed intelligence is not an afterthought, but a core adaptation for the Medusa stage. As the tide turns and currents strengthen, the jellyfish drifts back toward open channels between roots. Sunlight angles lower, casting elongated shadows that shift with the ripples. The Rhopalia continue their silent surveillance, each eye sampling fragments of the visual field, each processing hub contributing to the collective output. There is no central command issuing orders, yet the animal responds to its environment with precision that rivals organisms possessing complex brains. In the dense geometry of the mangrove forest, the box jellyfish demonstrates that intelligence need not reside in a single organ to be effective. By distributing perception and processing across multiple hubs, it achieves a form of awareness tailored to its translucent body and aquatic world. In this brackish nursery where river and sea converge, the box jellyfish moves with deliberate grace, avoiding roots, seeking prey, and maintaining orientation amid shifting light. Its existence challenges assumptions about the relationship between structure and function in nervous systems. Without a brain in the traditional sense, it navigates complexity, integrates sensory data, and executes coordinated responses. The guardian of these shallow corridors is brainless only in the narrowest anatomical definition. Functionally, it is a mosaic of neural centers operating in harmony, a reminder that evolution often solves problems not by building larger organs, but by reorganizing networks into forms uniquely suited to the demands of their environment. The continental shelf slopes gently into warm coastal waters, where currents sweep across sandy flats, broken by coral outcrops and scattered seagrass. Sunlight penetrates clean tropical seas in long, wavering shafts that illuminate suspended particles and cast rippling patterns across the bottom. From above, the seafloor appears deceptively uniform, a pale expanse punctuated by darker patches where stingrays settle, or where buried fish lie motionless beneath a thin veneer of sand. To most observers, it is a landscape defined by sight. To the Hammerhead Shark cruising just above the substrate, it is a field of invisible signals, a layered map of electrical disturbances and pressure gradients that reveal life concealed beneath the surface. The hammerhead silhouette is unmistakable. Its head extends laterally into a broad, flattened structure known as the cephalofoil, with eyes positioned at the far ends like sentinels on a wing. For years, speculation suggested that this peculiar shape functioned primarily as a hydrodynamic aid, perhaps improving maneuverability. While the cephalofoil does contribute to lift and allows for subtle banking turns, its most remarkable function lies not in steering, but in perception. Spread across the underside of the head are thousands of sensory pores connected to specialized electroreceptors, known as the ampullae of Lorenzini. These gelatin-filled canals detect minute electrical fields produced by the muscle contractions and nerve impulses of other organisms. By extending the head outward, the hammerhead increases the spatial separation between these sensors, effectively widening its sensory baseline. As the shark glides over the sand, its body remains steady, propelled by slow, powerful sweeps of its crescent-shaped tail. The pectoral fins angle slightly downward, maintaining a precise altitude above the substrate. The eyes, positioned laterally, provide a broad visual field, but it is the underside of the cephalofoil that performs the deeper scan. Every living fish, every buried ray, emits faint electrical signals as ions move across cell membranes. These signals are extraordinarily weak, measured in microvolts, yet in the conductive medium of seawater they propagate through short distances. The ampullae of Lorenzini are exquisitely sensitive, capable of detecting differences in electrical potential that are almost inconceivably small. The advantage of spreading these receptors across a wide head becomes evident when the shark passes over a hidden stingray. Buried beneath a thin layer of sand, the ray's spiracles pulse gently as it breathes, and its heart beats steadily within its body. The electrical fields generated by these activities extend upward into the water column, distorted slightly by the conductivity of sand and water. If the shark's receptors were clustered narrowly, distinguishing the direction of the source would be more difficult. Instead, the cephalofoil provides a stereo array. Electrical input reaching the left side of the head differs subtly in intensity and timing from that reaching the right. The shark's nervous system integrates these variations, constructing a three-dimensional impression of the field's origin. The process resembles triangulation. As the shark shifts slightly to one side, the relative strength of the signal changes across the head. By sampling these gradients continuously while swimming, the hammerhead refines its estimate of the prey's location. The sensory information does not exist in isolation. It is combined with input from the lateral line system, which detects water movement, and from olfactory organs that sample dissolved chemicals carried by currents. The cephalofoil, therefore, is not simply a broad platform for electroreception, but a multifunctional sensory array that integrates multiple modalities. The stingray beneath the sand remains still, relying on camouflage. Its flattened body blends seamlessly with the substrate, and its dorsal coloration matches the pale grains above. Many predators rely heavily on vision and might overlook such a concealed form. The hammerhead's approach is different. As it passes overhead, a slight deviation in electrical field strength registers across the cephalofoil. The shark banks gently, executing a tight turn that would challenge a species with a narrower head. The wide span of the cephalofoil generates lift as water flows across it, assisting in rapid directional changes without excessive energy expenditure. On the second pass, the electrical gradient sharpens. The shark lowers its head closer to the sand, amplifying the contrast between background noise and the prey's signal. The ampullae respond not only to the presence of an electrical field, but also to its oscillations, which correspond to the rhythmic contraction of muscles and the beating of a heart. In this sense, the hammerhead is perceiving life at its most fundamental level, detecting the ionic language of physiology itself. The shark pauses briefly, hovering with minimal tail movement, its body stabilized by subtle adjustments of its fins. Then, in a sudden downward thrust, it presses the cephalofoil against the sand. The impact is deliberate. By pinning the stingray with the broad head, the shark immobilizes its prey and prevents escape. The mouth, positioned on the underside of the head, opens to seize the ray. The ray's tail whips reflexively, but the shark's positioning has neutralized the advantage of concealment. What appeared to be empty sand moments before becomes the site of a precise extraction. The shark lifts slightly, jaws clamped, and shakes its head to tear flesh. The sand settles again, erasing the disturbance as if nothing had occurred. This hunting strategy reflects evolutionary specialization. Hammerhead sharks inhabit a range of coastal and offshore environments, and many species exhibit seasonal migrations linked to temperature and prey availability. In warmer months, they may aggregate in large schools near seamounts or reefs, possibly for social or reproductive purposes. In cooler seasons, individuals disperse along coastlines, following prey distributions shaped by ocean currents. Their physiology supports these movements. As ectothermic animals, their body temperature aligns with surrounding water, yet their large size and muscular activity can create slight thermal advantages that enhance swimming efficiency. The cephalofoil also influences vision. With eyes positioned at the far ends, hammerheads achieve a wide visual field that overlaps slightly in front, providing a degree of binocular vision. This configuration enhances depth perception directly ahead while maintaining extensive lateral awareness. In open water, such a field of view aids in detecting both prey and potential threats. When combined with electroreception, the shark operates with a layered sensory system that captures both the visible and the hidden. Juvenile hammerheads often inhabit shallow nurseries such as estuaries or protected bays, where predation risk from larger sharks is reduced. In these habitats, electroreception may assist in locating small fish and crustaceans within murky water where visibility is limited. As they mature and venture into deeper waters, their diet expands to include larger prey, including rays, smaller sharks, and bony fish. The cephalofoil grows proportionally, increasing the surface area available for sensory input. This ontogenetic change underscores the relationship between morphology and ecological niche. The ocean floor remains a dynamic mosaic of signals. Even in the absence of obvious prey, background electrical noise exists, generated by microbial activity, moving water, and geological features. The hammerhead's nervous system must filter this noise, distinguishing biologically relevant patterns from ambient fluctuations. The ampullae of Lorenzini connect to sensory neurons that relay information to specific brain regions dedicated to electroreception. These regions process spatial and temporal variations, allowing the shark to ignore static fields and focus on pulsing signals characteristic of living organisms. As the sun declines and shadows lengthen across the sand, the hammerhead continues its patrol. It does not rush. Energy conservation is crucial for a predator that may travel considerable distances in search of food. The rhythmic sweep of its tail propels it forward with steady efficiency. Beneath the calm surface, the cephalofoil remains active, sampling gradients invisible to any human observer. Each pass over the seafloor updates the shark's internal map, refining estimates of where life lies concealed. In this vast and seemingly open habitat, concealment offers no absolute safety. The hammerhead's adaptation transforms the ocean into a transparent medium for electrical perception. By spreading its electroreceptors across a wide head, it achieves a form of stereo detection that allows precise localization of prey buried beneath sand. The cephalofoil is not an ornamental oddity, but a finely tuned instrument, expanding the spatial resolution of a sensory system that reads the faint currents of life itself. In doing so, the hammerhead shark embodies a principle of marine evolution. When vision reaches its limits, other senses extend into the invisible, revealing a hidden world of signals waiting to be interpreted by those equipped to perceive them. In the shallow, tropical shallows, where the sea thins into a mosaic of turquoise flats and winding tidal channels, sunlight reaches the bottom with startling clarity. Beneath the rippling surface lies a vast underwater pasture, a living plain of seagrass anchored in sand and silt. These meadows are not algae, but flowering plants, descendants of terrestrial ancestors that returned to the ocean millions of years ago. Their blades sway in gentle arcs, trapping sediment, stabilizing the seabed, and converting sunlight into the base currency of coastal ecosystems. Juvenile fish weave through the foliage. Sea cucumbers process organic debris between the roots. Crabs patrol the edges where meadow meets open sand. It is a habitat defined by quiet productivity, where growth is incremental and grazing is constant. From the deeper channel at the meadow's edge, a broad grey shape emerges with deliberate calm. The dugong moves without haste, its paddle-shaped tail sweeping side to side in slow, powerful strokes. Unlike dolphins or whales that frequent open water, the dugong belongs to a lineage specialized for benthic life. It is one of the last surviving representatives of the Sirenians in this region, a marine mammal that has evolved to feed almost exclusively on seagrass. Its body is streamlined but heavy, with a rounded torso and a downturned snout that seems perpetually oriented toward the seabed. There is no rush in its movement because its feeding strategy depends not on speed, but on endurance. As the dugong approaches the meadow, it exhales softly at the surface and then sinks with minimal effort. Its descent is not abrupt. Instead, it lowers itself as though gravity exerts a stronger pull on it than on most marine mammals. This is no illusion. Dugongs possess unusually dense bones, particularly in the ribs and limb bones, a condition known as pachyostosis. The increased bone mass acts as biological ballast, counteracting the buoyant force of air-filled lungs and fat tissue. Where many marine mammals must actively adjust buoyancy with muscular effort, the dugong's skeletal density allows it to settle naturally toward the bottom. In effect, its skeleton functions like built-in scuba weights, anchoring the animal within the water column so it can graze without constant vertical correction. Once on the seafloor, the dugong tilts forward, positioning its snout directly over the seagrass. Its lips are thick and muscular, forming a prehensile structure capable of precise manipulation. Sparse bristles cover the upper lip, acting as tactile sensors that detect the texture and density of vegetation. The mouth is not designed for tearing large prey or seizing fish. Instead, it operates as a vacuum-like apparatus. The dugong presses its snout into the sediment and clamps its lips around a cluster of blades. With a powerful contraction of jaw muscles and a backward sweep of the head, it uproots entire plants, roots and all. Sand and silt plume upward in a cloudy burst, momentarily obscuring the scene. This feeding action leaves a distinct trail, a furrowed path where vegetation has been stripped from the substrate. Unlike turtles that clip blades at mid-length, dugongs often excavate, removing rhizomes embedded in the sediment. The efficiency of this method can resemble industrial harvesting. Over time, repeated grazing shapes the composition of the meadow. Dugongs show preference for certain seagrass species, particularly those with high nitrogen content and lower fiber. By selectively removing favored plants, they influence competitive dynamics within the meadow, allowing less preferred species to expand. In this way, the dugong acts as an ecological engineer, its appetite subtly reshaping plant communities and nutrient cycles. The cloud of sediment stirred by feeding does more than obscure vision. It releases trapped nutrients into the water column, potentially stimulating microbial activity and influencing local chemistry. Fish that follow behind the dugong seize invertebrates exposed by the disturbance, benefiting from the excavation. This secondary feeding by opportunistic species highlights the interconnected nature of the meadow ecosystem. What appears to be a solitary act of grazing reverberates outward, affecting organisms beyond the immediate interaction. The dugong's ability to remain submerged for extended periods enhances its grazing efficiency. Although it must surface periodically to breathe, it can spend many minutes underwater between breaths. Its lungs extend along much of its body length, contributing to buoyancy control and oxygen storage. The dense bones reduce the need for constant swimming effort to remain near the bottom, conserving energy. In warmer waters, thermoregulation becomes an important consideration. Unlike cetaceans with thick blubber layers adapted to cold seas, dugongs inhabit tropical and subtropical zones where overheating can be a risk. Their relatively modest fat layer reflects this balance, sufficient for insulation but not excessive. The shallow habitats they frequent can warm rapidly under intense sunlight, and dugongs may adjust feeding times to cooler periods of the day, moving into slightly deeper water when temperatures peak. A calf swims close to an adult female, mirroring her movements. Dugongs have long maternal bonds, and calves nurse for extended periods while gradually learning to forage. The downturned snout and lip structure are present early, but mastering efficient grazing requires practice. The calf experiments with small patches of seagrass, tugging clumsily at first. The mother continues her steady excavation, her tail occasionally brushing the calf as a subtle reassurance of proximity. Reproduction in dugongs is slow, with long gestation periods and typically a single calf born at a time. This low reproductive rate makes populations vulnerable to habitat degradation and human disturbance, particularly in coastal areas where boat traffic and fishing gear intersect with seagrass meadows. The meadow itself reflects seasonal rhythms. In some regions, monsoon cycles or shifts in freshwater input alter salinity and sedimentation patterns, influencing seagrass growth. Dugongs may undertake localized migrations in response to these changes, moving along coastlines to track areas of peak productivity. Their movements are not as dramatic as those of whales traversing entire ocean basins, yet they represent a form of spatial memory and environmental awareness. Individuals may return repeatedly to favored feeding grounds, suggesting familiarity with resource distribution. As the dugong continues grazing, the pattern of furrows expands. The meadow is not destroyed indiscriminately. Seagrass possesses remarkable resilience, capable of regrowing from remaining rhizome fragments. Moderate grazing can stimulate new growth, much like terrestrial herbivory shapes grasslands. The dugong's feeding thus exists within a feedback loop. By preventing overdominance of certain species and promoting turnover, it contributes to meadow health. However, excessive pressure, whether from large dugong populations or from additional stressors such as pollution and coastal development, can tip the balance toward decline. The delicate equilibrium between herbivore and habitat underscores the interconnectedness of benthic systems. From a distance, the dugong's form appears almost prehistoric, its lineage tracing back to a time when marine herbivory was more widespread. Fossil records reveal relatives that once inhabited diverse coastal zones across the globe. Today, the dugong's range is more restricted, its survival intertwined with the fate of seagrass ecosystems that themselves depend on clear water and stable sediment. Turbidity from dredging or runoff can block sunlight, reducing photosynthesis and weakening meadows. In such conditions, dugongs may be forced to travel farther between feeding sites, increasing energy expenditure and exposure to threats. As the tide begins to ebb, currents strengthen slightly, and the dugong lifts from the meadow with a slow sweep of its tail. The dense bones that facilitated descent now require steady propulsion to ascend. It rises to the surface, exhales in a soft plume, and inhales before sinking once more. The rhythm of feeding resumes, measured and methodical. Each uprooted plant, each sediment plume, is part of a larger ecological narrative. The dugong does not hunt with stealth or strike with speed. It excavates, transforms, and grazes with a persistence that shapes the seafloor itself. In this underwater pasture, the dugong embodies a different model of marine survival. Its adaptations are not weapons but tools for endurance. Heavy bones anchor it where food grows. Prehensile lips harvest vegetation with precision. Extended dives and careful buoyancy control maximize foraging efficiency. Through these traits, the dugong becomes a benthic excavator, a grazer whose presence is written across the meadow in furrows of sand and regrowth of grass. In the quiet shallows, where sunlight dances across blades and sediment drifts in lazy clouds, the dugong's slow movements reveal a strategy built not on conquest, but on sustained, intimate engagement with the seafloor that sustains it. In the silty estuaries of Southeast Asian coasts, where rivers exhale brown sediment into shallow tropical seas, visibility is rarely pristine. The seafloor is a soft plain of mud and broken shell, scored with burrows and rippled by faint tidal currents. Unlike coral reefs that advertise their inhabitants in color and motion, this habitat conceals its dramas beneath a veil of suspended particles. Here, outlines blur quickly. Shadows dissolve into substrate. Survival favors those who can disappear, not into brilliance, but into ambiguity. A narrow arm slips from a shallow hole in the mud. Then another. Slowly, the entire body of a mimic octopus emerges, lifting itself on flexible limbs as though testing the temperature of the water. Its mantle is pale beige, streaked with darker bands, a coloration that matches the mottled sediment around it. At first glance, it appears unremarkable, simply another small cephalopod navigating a dangerous neighborhood. But within seconds, its identity becomes unstable. Its body elongates, arms spreading and flattening as it glides just above the substrate. The change is not random camouflage. It is transformation with intent. The mimic octopus inhabits a world where predators are numerous and varied. Groupers patrol the edges of the mudflat. Lionfish hover above small ridges. Sea snakes move with slow, confident undulation across the bottom. Each predator responds to specific cues. Some are deterred by venomous patterns. Others rely on movement recognition. The mimic octopus has evolved not merely to hide from these threats, but to impersonate them. It is the only known animal capable of mimicking multiple different species in context-dependent ways, selecting which model to assume based on the nature of the threat. A predatory fish approaches from the open water, its silhouette dark against the hazy light. The octopus flattens its body and pulls its arms together, fusing them into a broad, leaf-like form. It presses close to the mud and begins to swim with lateral undulations that mirror those of a flatfish. The stripes along its body sharpen and realign, creating the illusion of fins along its edges. The resemblance is startling, not only in appearance, but in motion. Flatfish are known for their toxic or unpalatable status in some species, and predators accustomed to avoiding them may hesitate. In that hesitation lies opportunity. The octopus glides forward, maintaining the ruse until the threat passes. Moments later, a different disturbance ripples through the sediment. A sea snake enters the frame, its body banded in high contrast black and white. Sea snakes are venomous and command respect in these waters. The octopus reacts differently. It buries most of its body in the mud, leaving two arms extended in opposite directions. These arms stiffen and adopt the same banded pattern through rapid chromatophore activation. The remaining arms remain concealed beneath the sediment. From a distance, the extended limbs resemble two separate sea snakes emerging from the substrate. The predator approaching from another angle slows, recalculates, and veers away. These transformations occur within fractions of a second. The mimic octopus achieves this through a combination of extraordinary neuromuscular control and sophisticated visual processing. Like other cephalopods, it possesses a large brain relative to body size, with significant portions dedicated to vision and motor coordination. Its eyes detect contrast, movement, and spatial relationships with high acuity. Although color perception in octopuses differs from that of vertebrates, they can detect polarization and fine variations in brightness. When confronted with a predator, the octopus must rapidly identify the species and its likely behavioral patterns. This assessment informs which mimicry strategy will be most effective. The skin of the mimic octopus contains layers of chromatophores, iridophores, and leucophores, similar to those of cuttlefish, but applied here in service of impersonation rather than concealment alone. Chromatophores expand or contract under direct neural control, altering color and pattern instantly. Beneath them, Iridophores reflect specific wavelengths, adding structural color and sheen. Papillae, small muscular hydrostats embedded in the skin, can be raised to create texture, converting smooth surfaces into rough, spined or ridged forms. When imitating a lionfish, for example, the octopus spreads its arms outward and elevates papillae to simulate fin rays, while adjusting coloration to mimic the lionfish's distinctive stripes. Perhaps most remarkable is the absence of a rigid skeleton. The octopus body is supported by a hydrostatic system where muscles arranged in complex layers allow for extension, compression, bending, and twisting without bones. This flexibility permits radical reconfiguration. The mantle can elongate to resemble a fish body. Arms can be paired, fused, or stiffened to simulate tails or spines. The nervous system coordinates these changes at extraordinary speed. Motor neurons innervate chromatophores directly, allowing patterns to shift in milliseconds. The same neural network controls muscular hydrostats, reshaping the body with fluid precision. In the murky estuary, where visual cues are often degraded, motion becomes critical. Predators rely heavily on recognizing characteristic swimming styles. The mimic octopus does not merely change shape, it alters locomotion patterns. When imitating a flatfish, it performs synchronized arm undulations that produce lateral glide. When mimicking a sea snake, it moves only two arms in serpentine waves, while the rest remain hidden. These motor programs are distinct and must be stored, retrieved, and executed with timing that matches the model species closely enough to deceive observers. The cognitive demands of such mimicry suggest advanced neural integration. The octopus brain is organized into lobes that process visual input and coordinate arm movement. Unlike vertebrates, much of the octopus's nervous system resides within its arms, with local ganglia capable of semi-autonomous action. Yet in mimicry, global coordination is essential. Patterning must align with posture and movement simultaneously. This implies a hierarchical system where central processing selects a strategy and peripheral circuits execute fine motor adjustments. Beyond defense, mimicry may also play a role in predation. By resembling harmless species, the octopus can approach unsuspecting prey. Small fish accustomed to the presence of flatfish or other non-threatening organisms may ignore the disguised predator until it is too late. In this context, mimicry becomes an offensive tool. The octopus extends an arm slowly, suction cups poised, and captures a small crab distracted by sediment disturbance. The transformation back to a neutral form occurs seamlessly as the prey is drawn toward the beak concealed beneath the arms. The mimic octopus inhabits an ecosystem shaped by fluctuating tides and variable salinity. Estuarine waters can warm rapidly under tropical sun, influencing metabolic rates. As ectotherms, octopuses adjust activity levels in response to temperature shifts. Warmer water accelerates neural transmission and muscle contraction, potentially enhancing mimicry speed, but increasing energy demand. The octopus must balance risk and expenditure, selecting when to deploy elaborate displays and when simple camouflage suffices. Reproduction introduces vulnerability. Females guard eggs within burrows, reducing feeding and movement. During this period, mimicry may be limited, as remaining near the nest becomes paramount. After hatching, larvae enter the planktonic phase, drifting before settling into benthic habitats. Juveniles gradually refine mimicry through experience, suggesting learning plays a role alongside innate motor programs. Observations indicate that younger individuals may experiment with forms before achieving convincing resemblance. As dusk approaches, the estuary darkens. Suspended sediment reflects the last shafts of light, turning the water column into a diffuse glow. The mimic octopus pauses atop a slight rise in the mud, body returning to a mottled neutrality. For a moment, it is simply another invertebrate in a crowded habitat. Then a shadow passes overhead. In less time than it takes for that shadow to sharpen, the octopus elongates and stripes itself, becoming something else entirely. The living hologram of the mudflat does not rely on speed or venom. It survives through deception, refined to context. By processing visual information and converting it into immediate morphological transformation, it bridges perception and action with unmatched versatility. In a world where predators specialize and prey adapt narrowly, the mimic octopus has chosen a different path. It does not commit to a single disguise. It maintains a repertoire, selecting and deploying identities as circumstances demand. In doing so, it reveals that intelligence in the ocean is not always measured by dominance or size, but by the ability to read the environment and rewrite oneself within it in real time. In the protected lagoons where coral reefs soften into fields of seagrass and branching gorgonians sway in steady currents, the water column becomes a vertical tapestry. Sunlight filters through the surface in narrow beams, illuminating suspended plankton and outlining the silhouettes of fish weaving horizontally between stems. Most marine animals in this environment are oriented forward, built for streamlined propulsion through open water. Their bodies are designed around speed and escape, their heads leading the way as they cut through currents. But within this three-dimensional forest, another axis matters more than horizontal velocity. Among the upright blades of seagrass and the latticework of soft corals lives a creature that has redefined orientation itself. A seahorse hovers in profile, its body angled vertically as though gravity exerts a different rule upon it. The head bends forward at a distinct angle, the long snout pointing slightly downward, while the torso curves gently into a coiled tail wrapped firmly around a slender strand of vegetation. It does not swim with the decisive strokes of a fish. Instead, a small dorsal fin along its back vibrates at high frequency, producing a faint shimmer that propels it slowly upward or downward. Tiny pectoral fins near the head make subtle adjustments, correcting pitch and yaw with remarkable precision. Its posture appears fragile, almost ornamental, yet every aspect of its form reflects a structural adaptation to life among vertical surfaces. The tail is the anchor of this existence. Unlike the tails of most fish, which are flattened for propulsion, the seahorse's tail is prehensile. It can coil, grip, and hold with controlled force. This ability is rare among aquatic vertebrates and more commonly associated with certain land mammals, such as monkeys that grasp branches. In the seahorse, the tail is not round in cross-section, but squared, composed of a series of bony plates arranged in a segmented, interlocking architecture. These plates form a structure that is both flexible and resistant to crushing forces. Studies of the tail's geometry reveal that its square configuration distributes stress evenly, allowing it to bend without collapsing under pressure. When currents strengthen, or when a larger animal passes nearby, the seahorse tightens its grip, transforming vegetation into a living mooring post. This anchoring behavior is not merely a defensive tactic, it is integral to feeding. The seahorse is a sit-and-wait predator in an environment dense with microcurrents and drifting prey. Small crustaceans such as copepods and amphipods pulse through the water column, propelled by rhythmic strokes of their appendages. These movements generate minute vibrations that predators can detect. Conversely, many of these prey organisms are highly sensitive to disturbances in the surrounding water. The slightest pressure wave can trigger an escape response. For the seahorse, success depends on approaching without broadcasting its presence. The elongated snout is central to this strategy. It functions as a precision suction device, engineered to capture prey without creating detectable turbulence. When a suitable target drifts within range, the seahorse does not lunge forward in a dramatic strike. Instead, it remains anchored, body stable against the current. The head rotates with subtle alignment, guided by independently moving eyes that scan for motion and depth cues. In a fraction of a second, the seahorse expands the buccal cavity within its snout, creating a rapid but localized drop in pressure. Water and prey are drawn inward along a narrow channel. Because the opening is small and the movement highly controlled, the surrounding water experiences minimal displacement. To the prey, there is no advancing wall of pressure, no obvious hydrodynamic cue. The suction occurs so close and so swiftly that escape becomes improbable. This feeding method reflects a broader principle of stealth in fluid environments. Many predators create bow waves as they move, compressing water ahead of them. Such waves serve as early warning signals to sensitive prey. The seahorse's strategy circumvents this by decoupling body movement from feeding action. The body remains stationary, anchored by the tail, while only the head performs a short rotational motion. The actual capture event occurs within the snout's confined geometry. The efficiency of this low suction mechanism allows the seahorse to feed repeatedly throughout the day, consuming hundreds of small prey items to sustain its metabolism. Metabolic demands in seahorses are relatively high compared to their size. They lack a true stomach, and food passes quickly through the digestive tract. Continuous feeding becomes necessary. This requirement shapes their habitat preference. Seahorses favor areas with abundant microcrustaceans and sufficient vertical structure to provide attachment points. Seagrass meadows, mangrove roots, and coral branches serve this purpose. In these habitats, the interplay between plant growth and nutrient flow determines prey density. Healthy seagrass beds trap sediment and provide refuge for juvenile invertebrates. In turn, seahorses influence prey populations through persistent grazing, subtly participating in the trophic web. The vertical orientation of the seahorse body offers additional advantages. By aligning with upright vegetation, the animal minimizes its silhouette. Predators scanning horizontally may overlook a shape that mirrors the direction of stems and fronds. Camouflage further enhances this effect. Seahorses can alter their coloration over time, adjusting hues to match surroundings. While not as rapid or dramatic as cephalopod chromatophore displays, these changes allow integration into the background. Some species develop filamentous skin appendages that resemble algae, breaking up the outline even more effectively. Reproduction in seahorses introduces one of the most distinctive reversals in the animal kingdom. The male possesses a brood pouch on the underside of the abdomen, into which the female deposits eggs during courtship. Within this pouch, the male fertilizes and incubates the embryos. The pouch provides controlled salinity, oxygenation, and nutrient exchange, functioning in many ways like a placenta. Blood vessels lining the pouch facilitate gas transfer, and the male regulates osmotic conditions to match those of the surrounding water as embryos develop. This paternal investment alters the dynamics of mating systems and parental care. During gestation, the male continues to anchor and feed, balancing energy demands with the burden of developing offspring. As currents shift with tidal cycles, seahorses adjust their positions within the meadow. When flow intensifies, they select sturdier anchoring points, often grasping thicker stems or branching corals. Their dense, bony plates provide protection against minor impacts, and the segmented tail resists crushing if pressed between objects. The structural design of the tail not only facilitates grasping, but also acts as armor. Its square cross-section, compared to a round one, reduces the likelihood of catastrophic deformation under load. Engineers studying biomimetic materials have drawn inspiration from this configuration, recognizing its combination of flexibility and strength. Predation pressure remains constant. Larger fish patrol the meadow edges, and crustaceans may prey on juvenile seahorses. The anchored posture can be both asset and liability. While attachment reduces drift and hydrodynamic noise, it limits rapid escape. Seahorses compensate by blending into their surroundings and by selecting positions that offer visual obstruction. Their independently mobile eyes provide near panoramic coverage, allowing them to detect approaching threats without turning the body. The life cycle of a seahorse is closely tied to the health of coastal ecosystems. Pollution, habitat destruction, and overharvesting for traditional medicine or the aquarium trade threaten many species. Because seahorses rely on specific structural habitats and continuous prey availability, degradation of seagrass beds or coral reefs can lead to rapid population declines. Their vertical specialization, while evolutionarily successful in stable habitats, makes them vulnerable to environmental change. As the light shifts toward evening, plankton density increases slightly with the cooling surface layer. The seahorse tightens its tail around a swaying stem and continues its patient surveillance. Tiny crustaceans drift past, unaware of the narrow snout aligned with their path. The dorsal fin flickers invisibly, maintaining balance. In this vertical world, success does not come from speed or dominance. It comes from structural innovation and hydrodynamic subtlety. The seahorse embodies an elegant solution to life within structured habitats. Its prehensile tail anchors it against currents, transforming plants into fixed platforms. Its snout captures prey without betraying motion through disruptive waves. Its body orientation aligns with environmental geometry, minimizing detection. In the quiet oscillation of seagrass and the soft murmur of tidal flow, the vertical specialist demonstrates that survival in complex ecosystems often depends on redefining how a body moves through space. By turning upright and grasping rather than racing, the seahorse occupies a niche few others can exploit, revealing the power of specialization when anatomy, behavior, and habitat converge in precise harmony. In the shallow coastal margins of the Western Pacific, where sandy plains stretch beyond the shelter of coral reefs, the seabed appears at first glance uniform and empty. Fine grains shift with each passing swell, forming ripples that reflect the physics of tide and wind. Currents sweep across this open expanse in gentle but persistent streams, carrying plankton, detritus, and drifting larvae. The water above is clear and bright, and the light reaches the bottom in soft gradients that emphasize every contour in the sand. It is an environment shaped by motion, where stability is temporary, and any structure carved into the substrate is at risk of erasure. Yet in this shifting landscape, a pattern emerges that defies expectation. Across the sandy plain, a circular design takes form. Radiating ridges extend outward from a central depression, evenly spaced and symmetrically aligned. Between the ridges, shallow valleys curve in precise arcs, forming a pattern so deliberate that it resembles a crafted artifact rather than a natural feature. The structure measures several meters in diameter, its edges crisp against the otherwise featureless seabed. No storm has sculpted this geometry. No coral has anchored it. Instead, the architect is a small pufferfish, a species whose body is compact and unassuming compared to the monumental scale of its construction. The male pufferfish hovers just above the sand, his fins vibrating in rapid, controlled pulses. His body is modest in length, yet the pattern he creates spans many times that distance. With each movement, he dips his snout and uses subtle sweeps of his fins to push sediment into ridges. The process is meticulous. He swims outward from the center in a circular path, carving grooves with measured precision. The dorsal and pectoral fins act not only as propulsive organs, but as sculpting tools, generating localized currents that displace sand without disturbing the overall form. Over days of persistent effort, the pattern expands, each ridge refined until symmetry emerges from chaos. At first glance, this elaborate circle appears purely ornamental, a display designed to attract a mate. Indeed, the geometry plays a role in courtship. Females inspecting the area assess the precision and scale of the construction, selecting males whose circles demonstrate persistence and structural clarity. However, beneath the aesthetic dimension lies functional engineering. The radiating ridges alter water flow across the sandy bottom. In an environment where even modest currents can dislodge eggs or bury them beneath sediment, controlling hydrodynamics becomes essential. As tidal flow moves across the plain, water encounters the raised ridges and is redirected along the grooves toward the center. The ridges slow the velocity of water within the circle, reducing shear stress at the central depression. Fine particles carried by the current settle within this protected basin, creating a slightly firmer substrate. When the female eventually deposits her eggs in the central zone, they rest within a microhabitat buffered from direct current force. The circle functions as a hydraulic trap, shaping the boundary layer of water in a way that stabilizes the developing embryos. The male's role extends beyond construction. After mating, he remains near the circle, fanning the eggs with his fins to maintain oxygenation. This behavior prevents stagnation within the protected center. The design's geometry ensures that incoming water is slowed but not eliminated, allowing controlled exchange without turbulence. In this interplay between structure and movement, the pufferfish demonstrates an intuitive understanding of fluid dynamics, achieved not through conscious calculation, but through evolutionary refinement. The pufferfish belongs to a family renowned for defensive adaptations. When threatened, many species inflate their bodies by ingesting water, expanding into a near-spherical form that deters predators. The inflation is facilitated by specialized elastic skin and a reduced number of rigid ribs, allowing dramatic volumetric increase without structural damage. Spines that lie flat against the body in normal conditions become erect upon inflation, adding a physical barrier. Some species also harbor tetrodotoxin, a potent neurotoxin concentrated in organs such as the liver and ovaries. This toxin interferes with sodium channels in nerve cells, potentially causing paralysis in predators that attempt ingestion. The combination of physical expansion and chemical defense makes the pufferfish a formidable adversary despite its small size. In the context of the geometric circle, however, the defensive strategy shifts from self-protection to offspring safeguarding. Eggs laid on open sand would be vulnerable to predation and displacement. The male's construction modifies the environment to reduce these risks. Small invertebrates that might prey on eggs encounter the ridges as obstacles, and fish swimming across the plain are less likely to pass directly over the central depression. The circular form also creates visual contrast against the otherwise uniform substrate, possibly signaling occupancy to conspecifics and deterring rival males from encroaching. The precision of the design raises questions about sensory guidance. How does the pufferfish maintain symmetry over such a large scale relative to its body size? Visual cues likely play a central role. The fish's eyes, positioned laterally, provide a wide field of view that encompasses the entire circle as it swims around the perimeter. Subtle adjustments in fin stroke angle allow corrections to ridge height and spacing. The fish may also rely on tactile feedback, sensing resistance changes as sediment accumulates. The construction process unfolds gradually, with iterative refinement. Each pass reinforces existing ridges, smoothing irregularities and sharpening boundaries. Environmental conditions influence the success of the circle. Strong storms can erase weeks of effort in hours, flattening the sand and dispersing sediment. In such cases, the male must begin anew, expending energy that could otherwise support feeding or territorial defense. The persistence required for repeated construction underscores the selective pressure driving this behavior. Only males capable of sustained effort and precise motor control produce circles that withstand moderate current shifts and attract females. The sandy plain itself is a dynamic system. Burrowing organisms such as worms and small crustaceans churn the substrate, altering grain distribution. Microalgae colonize surface particles, binding them slightly and affecting cohesion. The pufferfish's construction interacts with these processes. By elevating ridges and deepening grooves, it redistributes sediment layers, potentially influencing microbial activity and nutrient cycling at a small scale. The circle becomes not just a courtship display, but a micro-ecosystem, with altered flow patterns and sediment characteristics that persist until natural forces reclaim the design. As the eggs develop within the central depression, the male's vigilance intensifies. He patrols the perimeter, confronting small fish that venture too close. If threatened directly, he may inflate, transforming from a sculptor into a defensive orb. Inflation increases body size dramatically, making ingestion difficult for gape-limited predators. The energy cost of repeated inflation is significant, as it requires active pumping of water into the stomach and subsequent expulsion. Thus, the male reserves this tactic for genuine threats, relying otherwise on proximity and motion to deter intruders. The geometric precision of the circle has drawn attention beyond the marine environment. To human observers, it evokes comparisons to land-based crop circles or engineered structures, yet its origin lies entirely in biological necessity. The scale of the circle relative to the fish's body amplifies its visual impact, creating an impression of disproportionate ambition. In truth, the design represents an evolutionary compromise between energy investment and reproductive success. A larger circle may offer greater hydraulic stability and increased visibility to females, but it demands more time and effort to construct and maintain. As currents shift with the turning tide, water glides over the ridges and into the central basin. Fine particles settle, and the eggs remain cradled within the shallow depression. The male hovers nearby, fins oscillating in small arcs that maintain position without disturbing the delicate structure. The circle stands as both advertisement and fortress, geometry imposed upon fluidity. In this open, sandy habitat, where permanence is rare and structures are transient, The pufferfish demonstrates a remarkable intersection of art and engineering. Its circular construction is not mere decoration, but a hydrodynamic adaptation shaped by the demands of reproduction. By altering current flow and stabilizing sediment, the fish enhances the survival prospects of its offspring. The same species that inflates into a spiny sphere and carries potent toxins reveals here a different facet of its biology. Patient sculptor, hydraulic strategist, and guardian of a fragile central cradle. In the interplay between moving water and shifting sand, the geometric artist inscribes purpose into a landscape otherwise defined by impermanence. The Antarctic shoreline does not begin gently. It fractures from ice into ocean with a violence that appears permanent, yet shifts daily with wind and tide. Sea ice groans and flexes along its edges, and katabatic winds spill from the interior continent like invisible avalanches of cold air. The water here is steel blue and mercilessly clear, revealing nothing of its depth except darkness. Beneath the surface, however, life concentrates in explosive abundance. Nutrient-rich currents rise from the continental shelf, feeding blooms of phytoplankton that in turn sustain krill, fish, and squid. It is a food web built on seasonal light, compressed into months of furious productivity. Along the ice edge, a colony of penguins gathers, their black and white bodies stark against the snow. They stand upright on land, but their design does not belong to the ice. It belongs to the water. A penguin steps toward the lip of the sea ice, peers down, and commits. The fall is abrupt. It hits the water and vanishes beneath the surface, transforming instantly from a waddling bird into a streamlined predator. On land, its gait appears awkward, almost comic, but in water it becomes precise and economical. The wings, modified into rigid flippers, sweep in coordinated arcs, generating lift and thrust simultaneously. The body is torpedo-shaped, tapering toward the feet, which act as stabilizers. Within seconds, it is descending through a column of frigid water, leaving behind only a brief ripple on the surface. The secret to this transition lies not only in musculature and bone structure, but in feathers. Unlike most birds, penguins possess densely packed short feathers layered over one another in a tightly interlocking arrangement. Each feather is stiff and narrow, creating a smooth outer surface that resists water penetration. Beneath this outer layer lies a thick down layer that traps air close to the skin. On land, this trapped air functions as insulation, forming a thermal barrier that reduces heat loss in sub-zero conditions. In water, it plays an additional role that reshapes the physics of swimming. As the penguin dives deeper, pressure increases rapidly. The trapped air compresses against the body, reducing buoyancy and allowing easier descent. But the air does not disappear. It remains lodged between feathers and skin, forming a microlayer that clings to the body surface. At certain depths, the penguin begins to release this air deliberately. Tiny bubbles stream along its flanks, racing toward the tail. The effect is subtle but profound. The bubbles form a lubricating layer between the bird and the surrounding water, reducing drag by altering the boundary layer where water molecules interact with the body. Hydrodynamics in water are unforgiving. Water is nearly 800 times denser than air, and moving through it requires overcoming substantial resistance. Friction between body surface and water creates drag that limits speed and increases energy expenditure. By releasing microbubbles in a controlled manner, penguins reduce this friction. The bubbles act as a thin cushion, decreasing the viscosity effect at the surface and allowing water to slip past more easily. In essence, the penguin becomes a bubble-lubricated torpedo, accelerating with efficiency that would otherwise demand disproportionate muscular effort. The timing of bubble release is not random. Observations reveal that penguins often release more bubbles during ascent than descent. As they complete a hunting dive and begin rising toward the surface, buoyancy increases as pressure decreases and trapped air expands. The bird capitalizes on this by releasing compressed air in bursts, gaining both lift and reduced drag simultaneously. This combination allows acceleration to remarkable speeds. Measurements from biologging devices have recorded bursts that exceed what would be expected based on muscle power alone. The lubrication effect and expanding buoyancy create a compound acceleration, propelling the penguin upward in what researchers describe as porpoising exits. But before ascent comes the hunt. Below the ice, schools of Antarctic silverfish shimmer in loose formations, their bodies reflecting faint light filtering from above. Krill swarm in dense clouds, their translucent forms pulsing as they feed on phytoplankton. The penguin's eyes, adapted for underwater vision, adjust to the dim environment. The cornea, important for refracting light in air, becomes less effective in water due to similar refractive indices, so the penguin relies more heavily on the lens for focusing. Muscles adjust lens shape rapidly, allowing clear vision at varying depths. The bird's retina contains photoreceptors tuned to detect contrast in low light, essential for tracking prey against the darkening water. As the penguin approaches a school of fish, it alters wing stroke frequency. The flippers beat in short, powerful arcs, steering with subtle adjustments of head and feet. Unlike many fish that rely on lateral body undulation, penguins generate thrust through oscillating hydrofoils. This method produces lift on both the downstroke and upstroke, maximizing propulsion. The body remains rigid and aligned with the direction of travel, minimizing turbulence. A fish veers away at the last moment, sensing pressure changes in the water. The penguin adjusts course instantly, releasing a thin stream of bubbles that trails along its side. The microbubble layer reduces drag, allowing a sharper turn without energy loss. The fish darts again, but the penguin closes the distance. Its beak snaps shut around the prey, and it continues forward momentum before swallowing. There is no pause. In these icy waters, foraging efficiency determines survival. Energy spent must be balanced by caloric intake sufficient to maintain body temperature and fuel reproduction. Thermoregulation is an ongoing challenge. The surrounding water hovers near freezing, and heat loss can be rapid. The penguin's body counters this with a combination of insulation and vascular control. The dense feather layer traps air, while a thick layer of subcutaneous fat provides additional insulation. Beneath the skin, countercurrent heat exchange systems in the flippers and legs conserve core temperature. Arteries carrying warm blood from the body core run adjacent to veins returning colder blood from extremities. Heat transfers from artery to vein before reaching the flippers, reducing heat loss into the water. This system allows peripheral tissues to cool while preserving warmth in vital organs. As the penguin descends further, oxygen management becomes critical. Penguins possess high concentrations of myoglobin in their muscles, storing oxygen directly within tissue. Their blood volume relative to body size is also elevated, carrying more hemoglobin for oxygen transport. During dives, heart rate decreases, conserving oxygen by limiting circulation to non-essential tissues. The brain and muscles receive priority, while peripheral circulation diminishes. This dive response enables extended submersion without immediate need for breathing. When the hunt concludes and ascent begins, the compressed air layer between feathers expands rapidly. The penguin releases it in a cascade of tiny bubbles that stream past its body like silver threads. The friction reduction intensifies, and buoyancy adds to propulsion. The bird accelerates upward, breaking through the surface in an explosive burst. Water sheds instantly from its plumage due to the feather microstructure, which resists wetting. The penguin arcs briefly into air before re-entering, a maneuver known as porpoising. This technique allows rapid travel at the surface while minimizing drag, alternating between swimming and leaping. On the ice edge, other penguins observe the return. Some will dive next, repeating the cycle. The colony's survival depends on collective efficiency. During breeding season, adults must commute between feeding grounds and nests, sometimes traveling considerable distances across shifting pack ice. The bubble lubrication effect enhances not only individual hunting success, but also overall energy economy during these commutes. In a region where storms can arise suddenly and sea ice conditions change unpredictably, conserving energy can mean the difference between reaching the colony with sufficient food for chicks or returning depleted. The Antarctic ecosystem magnifies small advantages. Predators such as leopard seals patrol beneath ice floes, scanning for silhouettes against the light. Penguins reduce vulnerability by entering and exiting water in coordinated bursts, minimizing time spent at the surface where seals attack. The bubble lubrication that aids acceleration also shortens exposure windows, allowing rapid entry into deeper, safer water layers. As twilight spreads across the ice and the sea darkens to indigo, the colony settles into relative calm. Beneath the surface, however, the drama continues. Penguins dive again and again, their bodies sheathed in microscopic air, transforming friction into opportunity. The adaptation that began as insulation against cold has evolved into a dynamic hydrodynamic tool. In this environment of ice and darkness, where physics imposes relentless constraints, the bubble-lubricated torpedo redefines what is possible. Through the controlled release of trapped air, penguins bend fluid resistance to their advantage, accelerating beyond expectations and turning the unforgiving density of water into a medium they command with astonishing precision. Across the coral reef, flat at low tide, the water lies only waist-deep above a landscape that appears still and uncomplicated. Branching corals extend upward like brittle antlers. Massive boulder corals sit in quiet permanence, their surfaces patterned with polyps that retract as shadows pass overhead. Between these calcified structures, the reef floor is layered with sand, fragments of broken shell, and fine organic detritus that settles in crevices where currents slow. In this overlooked layer, beneath the spectacle of fish and corals, lies an animal that appears inert to casual observation. It is elongated, thick-skinned, and slow to the point of invisibility. The sea cucumber does not dart, does not flash color, and does not chase prey. Yet within its soft body exists one of the most remarkable material systems in the animal kingdom. The sea cucumber moves almost imperceptibly along the substrate, its tube feet extending and retracting in coordinated waves beneath its body. To the eye, it resembles a pliable sack of tissue, mottled brown or gray, blending into the sediment it processes. It is an echinoderm, related to sea stars and sea urchins, but unlike its armored relatives, it has surrendered rigid plates in favor of a flexible form. This flexibility is not merely softness, it is dynamic material control at the molecular level. Within its body wall lies a network of collagen fibers embedded in a mutable connective tissue that can alter stiffness on demand. Through chemical signaling, the sea cucumber can transform this tissue from compliant and fluid-like to rigid and resistant within seconds. As it approaches a narrow crevice between two coral heads, the sea cucumber elongates slightly, pressing the front of its body against the gap. The opening appears too small to accommodate its bulk, yet the animal does not retreat. Instead, its body begins to soften. The collagen fibers within its connective tissue disengage from cross-linking bonds that normally provide structural integrity. As these bonds relax, the tissue loses stiffness, becoming pliable and deformable. The sea cucumber effectively liquefies its posture without losing cohesion. It flows into the crack, compressing and elongating simultaneously, guided by subtle contractions of longitudinal and circular muscles embedded within the body wall. This capacity for collagen manipulation is controlled by neurochemical signals. Specialized cells release factors that influence the bonding between collagen fibrils, altering mechanical properties without requiring structural rebuilding. In engineering terms, the sea cucumber operates a biological smart material system, adjusting stiffness through reversible cross-linking. When the animal has squeezed fully into the protective crevice, it reverses the process. Crosslinks reform, collagen fibers lock into place, and the body wall stiffens dramatically. In this hardened state, the sea cucumber can resist predatory attempts to extract it from its refuge. The once soft, yielding body becomes firm, comparable in resistance to rigid organic materials. This transformation is not purely defensive. It is integral to daily survival on a reef where spatial competition is intense. Crevices provide shelter from fish that might otherwise nip at exposed tissue. They also buffer against currents and temperature fluctuations. During low tide or in shallow reef flats, where water warms rapidly under tropical sun, retreating into shaded cracks can reduce thermal stress. Although sea cucumbers are ectothermic and cannot regulate internal temperature metabolically, their behavior mitigates exposure to extremes. The ability to liquefy and infiltrate tight spaces expands the range of microhabitats they can exploit. Yet the sea cucumber's ecological significance extends beyond material manipulation. It is a detritivore, a recycler of organic matter that accumulates between corals. With a crown of branching tentacles surrounding its mouth, it gathers sediment and ingests it in steady, rhythmic motions. The tentacles are coated in mucus that traps organic particles, including microalgae, bacteria, and decomposing material. Once inside the digestive tract, nutrients are absorbed and the remaining sediment is excreted as processed sand. This continuous ingestion and digestion aerates the substrate, influences microbial communities, and alters nutrient availability. In effect, the sea cucumber functions as a benthic mechanic, maintaining the health of reef sediments through persistent turnover. Predators do not ignore such a nutrient-rich body. Fish, crabs, and even some sea stars may attempt to feed on sea cucumbers. When threatened, certain species deploy another unusual defense: evisceration. They can expel portions of their internal organs, including sticky threads known as Cuvierian tubules, through the anus. These tubules entangle attackers and may contain toxic compounds that deter further assault. After evisceration, the sea cucumber regenerates lost tissues over weeks. This capacity for regeneration reflects a broader theme in echinoderm biology: resilience through modularity. Organs can be sacrificed and rebuilt, and connective tissues can be softened or hardened as needed. The interplay between collagen manipulation and regeneration reveals a sophisticated balance between vulnerability and control. A body that can become fluid risks mechanical damage if not carefully regulated. The sea cucumber's mutable connective tissue is stabilized by a matrix of proteoglycans and interfibrillar molecules that prevent complete structural collapse. Muscle fibers maintain basic alignment even when stiffness decreases, ensuring that the animal remains coherent rather than disintegrating. When stiffening occurs, bonds between collagen fibrils increase tensile strength, distributing force across the body wall. In the broader reef ecosystem, sea cucumbers influence sediment chemistry in ways that ripple outward. By processing sand and releasing ammonia and other nutrients, they enhance conditions for microbial communities that in turn support benthic food webs. Areas with high sea cucumber density often exhibit altered sediment composition compared to zones where they have been removed. Overharvesting of sea cucumbers for culinary markets has led to measurable changes in reef health in some regions, underscoring their functional importance. As dusk approaches and reef fish retreat to shelter, the sea cucumber remains active. Its tube feet grip the substrate, and its body undulates slowly forward. The surface texture, covered in small papillae, assists in sensing contact and detecting pressure changes in the surrounding water. Although lacking complex eyes, sea cucumbers possess light-sensitive cells that help them orient relative to surface brightness. This sensory input informs movement towards shaded refuges during intense daylight. Within its elongated form lies a water vascular system inherited from its echinoderm ancestry. This system, a network of fluid-filled canals, powers the extension of tube feet through hydraulic pressure. The coordination between hydraulic control and collagen manipulation allows for fine-tuned movement and anchoring. When the animal stiffens its body wall within a crevice, it often reduces tube foot extension, relying on hardened tissue to maintain position rather than active muscular grip. This reduces energy expenditure while maximizing protection. Occasionally, a predatory fish probes a crack with its snout, attempting to extract hidden prey. If the sea cucumber senses persistent pressure, it may stiffen further, resisting removal. Should extraction become imminent, it may resort to evisceration, sacrificing expendable tissue to escape. The regrowth process that follows requires metabolic resources and time, yet the trade-off favors survival over immediate structural integrity. As currents shift with the turning tide, fine particles settle once more across the reef floor. The sea cucumber emerges from its crevice, body gradually softening as it resumes foraging. The transformation from rigid defender to pliable grazer occurs seamlessly, without visible markers of transition. It is an internal negotiation of bonds and forces, orchestrated at the microscopic level. In the language of materials science, the sea cucumber embodies adaptive stiffness. In ecological terms, it represents quiet maintenance. It neither hunts nor grazes on living coral, yet its role is foundational. By turning sediment into renewed substrate, and by altering its own mechanical state to navigate and survive, it operates as a liquid mechanic within the reef. In a habitat often celebrated for color and speed, the sea cucumber demonstrates that resilience can reside in softness, and that mastery over one's own molecular architecture can be as decisive for survival as claws or teeth. Along the outer edge of a tropical reef, where coral gives way to rubble and sand channels weave between outcrops, the seafloor resembles a patchwork quilt of living and broken matter. Fragments of coral skeleton lie scattered among sponges and tunicates, and patches of macroalgae sway in response to passing swells. Small fish hover in loose schools above the substrate, dipping down to investigate crevices before retreating to open water. The environment is not static, but constantly rearranged by currents, grazing, and predation. Within this shifting mosaic, certain organisms have learned not only to survive among the reef's diversity, but to carry a portion of it with them. A small crab moves across the rubble field with careful deliberation. Its legs are long and spindly, lifting its compact body just high enough to clear irregularities in the terrain. At first glance, it does not appear as a distinct animal at all. Its carapace is obscured beneath an uneven layer of algae, sponge fragments, and the pale tentacles of small sea anemones that ripple gently in the water. Only when it pauses and adjusts its footing does the illusion dissolve, revealing the subtle articulation of joints and the slow pivot of stalked eyes scanning the surroundings. This is a decorator crab, a crustacean that has transformed camouflage into a form of active ecological engineering. The decorator crab's strategy is not merely to blend in with its environment, but to physically incorporate elements of that environment into its own structure. Its exoskeleton is covered with specialized hooked setae, microscopic bristle-like projections that curve at their tips. These hooks function much like biological Velcro, capable of gripping fibrous or soft materials pressed against them. When the crab encounters suitable organisms such as sponges, hydroids, algae, or sea anemones, it uses its chelae to pluck small pieces and carefully press them onto its carapace. The hooked setae secure these additions in place, anchoring living tissue to the crab's shell. In this process, the crab becomes a mobile substrate, a platform upon which other organisms continue to live and grow. The selection of materials is not random. Decorator crabs often show preference for organisms that provide defensive benefits, particularly those with chemical deterrence or stinging capabilities. Sea anemones, with their tentacles armed with nematocysts, are especially valuable. By transplanting anemones onto its back, the crab equips itself with a perimeter of stinging cells capable of deterring fish or octopuses that might otherwise attempt predation. The relationship between crab and anemone is more complex than simple attachment. Once secured to the carapace, the anemones remain alive, their tentacles extending outward to capture plankton and organic particles. The crab's movement through the reef exposes them to new feeding opportunities. In return, the anemones provide both camouflage and active defense. When a predator approaches, the tentacles brush against its sensitive skin or gills, delivering stings that encourage retreat. In this sense, the crab has created a symbiotic partnership, relocating an organism that would normally be fixed to rock and granting it mobility. Maintaining such a mobile ecosystem requires ongoing care. As the crab grows, it periodically molts, shedding its old exoskeleton to accommodate increased size. Before molting, the crab may transfer some of its decorative organisms from the old shell to the new, softer one. This behavior suggests recognition of the functional value of its attachments. The process is delicate. The crab must remove anemones or sponge fragments without causing fatal damage, then secure them onto fresh setae before the exoskeleton hardens. During this vulnerable period, when the new shell is soft and the decorations are not yet fully secured, the crab faces heightened predation risk. The exoskeleton itself is a composite of chitin and calcium carbonate, forming a rigid external skeleton that protects internal organs and provides attachment points for muscles. The hooked setae are embedded within this structure, arranged in patterns that maximize surface area for attachment. Unlike smooth-shelled crabs that rely solely on coloration for concealment, decorator crabs externalize camouflage into 3 dimensions. The added organisms alter the crab's outline, breaking up recognizable shapes and blending it into the heterogeneous reef background. As the crab advances across the substrate, it encounters a patch of filamentous algae. With deliberate motions, it trims a small piece and presses it onto a bare section of carapace. The algae's fine strands entangle within the hooked setae, securing the fragment. Over time, such additions can grow, further obscuring the crab's form. In areas with high predation pressure, decorator crabs may accumulate denser coverings, investing more energy into maintenance. In relatively sheltered zones, they may adopt lighter decoration, conserving effort for foraging and reproduction. Predator interactions reveal the effectiveness of this strategy. A wrasse approaches, investigating potential prey among rubble. The crab freezes, legs tucked slightly inward, minimizing motion that might reveal its identity. The fish's attention shifts to a nearby worm, ignoring the crab entirely. In other instances, when motion is detected and a predator makes contact, the stinging tentacles of transplanted anemones respond. Nematocysts discharge upon mechanical stimulation, injecting toxins into the predator's tissue. The discomfort may be mild or significant depending on species, but the sudden sting is often sufficient to discourage further probing. The decorator crab also feeds amid this camouflage. It scavenges detritus, consumes small invertebrates, and occasionally nips at organic material. The decorations do not impede feeding but rather provide cover as it searches. In some cases, the crab may even consume fragments of its own decorative organisms if resources are scarce, illustrating the fluid boundary between partnership and opportunism in reef ecology. This strategy has broader ecological implications. By relocating sessile organisms such as anemones or sponges, decorator crabs contribute to the dispersal of these species. When the crab moves to a new area and eventually molts, some attached organisms may detach and establish themselves on nearby substrate. In this way, the crab acts as a vector for colonization, influencing spatial distribution patterns across the reef. Environmental conditions shape decoration choices. In zones dominated by chemically defended sponges, crabs may favor those over anemones. In areas where algal cover is abundant, camouflage may rely more heavily on plant material. The crab's sensory system likely guides these decisions. Chemoreceptors on the antennae and mouthparts detect chemical cues indicating potential deterrent properties. Visual assessment of color and texture may also play a role, though underwater lighting conditions complicate perception. The decorator crab's behavior reflects an evolutionary pathway where morphology alone was insufficient for survival. Rather than developing thicker armor or faster escape responses, it outsourced part of its defense to other organisms. This outsourcing required structural innovation in the form of hooked setae and behavioral sophistication in selecting and maintaining attachments. It also demanded tolerance of living organisms on its body, navigating potential competition for space or resources. As dusk settles over the reef, plankton density increases and small fish retreat to crevices. The decorator crab continues its quiet patrol, its back a miniature reef in motion. The anemones atop its shell extend tentacles to capture passing particles, benefiting from the crab's mobility. The algae absorb faint light even as it fades, and the sponge fragments filter water around them. The crab's exoskeleton has become a scaffold for a living assemblage. In this layered ecosystem, the decorator crab exemplifies a form of ecological integration rarely seen with such physical intimacy. It is not merely camouflaged, but transformed into a composite organism, part crustacean, part transplanted reef. Through the simple mechanism of hooked setae and deliberate selection, it constructs a mobile ecosystem that enhances its survival while subtly reshaping the distribution of other species. The reef is often described as a community of interdependence, but here that interdependence is literal, bound to a shell and carried across the seabed on articulated legs. The reef slope drops gradually from a bright wave-washed crest into a quieter zone, where sand settles between coral heads and sponges rise like muted towers from the substrate. Light here is softer, filtered by depth and by the layered complexity of life above. Algae form small meadows on broken coral rubble, and scattered ascidians cling to rock faces like folded fabric. The water is calm but not still. Fine particles drift through the column, revealing the direction of subtle currents that sweep nutrients along the reef face. In this muted landscape, movement is usually obvious. Fish glide in arcs, parrotfish scrape coral, wrasses dart in quick bursts between shadows. But near the bottom, pressed against the substrate and barely distinguishable from the sponges it resembles, a frogfish waits. At first glance, it appears less like a fish and more like a growth on the reef. Its body is bulbous and irregular, covered in textured skin that mirrors the rough surfaces around it. Colors vary widely depending on species and environment, from mottled yellows and greens to dark browns speckled with white. Some individuals bear skin filaments that resemble algae or sponge fibers, breaking up their outline into organic fragments. Unlike streamlined reef fish designed for speed, the frogfish's form seems awkward, almost unfinished. Yet this is not a failure of hydrodynamic design. It is the deliberate abandonment of swimming as the primary means of locomotion. The frogfish does not cruise through the water column. Instead, it walks. Its pectoral fins are not thin, oscillating paddles, but stout, jointed structures that function more like limbs than fins. Supported by strong fin rays and muscular bases, they bend and press against the seafloor, lifting and shifting the body in short, deliberate steps. The pelvic fins contribute as stabilizers, forming a tripod with one pectoral fin while the other advances. This gait is slow but precise, allowing the frogfish to reposition itself without creating conspicuous water disturbances. In an environment where prey are sensitive to pressure waves, minimizing hydrodynamic noise is essential. The evolutionary shift from swimming to walking reflects a fundamental ecological decision. Frogfish are ambush predators. They do not pursue prey across open water, but rely on deception and proximity. By remaining close to the substrate and moving in increments that resemble drifting debris, they avoid detection by both prey and predators. Their skin contains chromatophores capable of adjusting coloration over time, though not with the rapidity of cephalopods. Changes occur over days or weeks, aligning the frogfish's appearance with surrounding sponges or corals. Texture adds another layer to this camouflage, with small dermal appendages mimicking encrusting growth. As the frogfish advances across a patch of coral rubble, a small goby hovers nearby, scanning for plankton. The goby's lateral line system is tuned to detect water displacement caused by approaching threats. A fast-swimming predator would generate a bow wave, alerting the goby before contact. The frogfish avoids this entirely. Its walking motion displaces little water. Each step is controlled and deliberate, shifting the body by centimeters rather than meters. The frogfish pauses frequently, blending into the background so effectively that even a trained observer might overlook it. The head is oversized relative to the body, dominated by a cavernous mouth that seems disproportionate to the fish's bulk. This mouth is not merely large, it is an expansion device capable of engulfing prey nearly equal in size to the frogfish itself. The jaws hinge open with astonishing speed, and the buccal cavity expands dramatically. When the strike occurs, it unfolds within milliseconds. Water and prey are sucked inward in a single explosive motion. The acceleration of water into the mouth is so rapid that it creates a vacuum effect, drawing prey in before it can react. To bring prey within range, the frogfish employs a specialized lure known as the elysium, a modified dorsal fin spine that extends forward above the mouth. At its tip sits a fleshy appendage called the esker, often shaped and colored to resemble a small worm or shrimp. The frogfish twitches this lure in subtle, irregular movements, simulating the erratic motion of prey. Small fish attracted to the apparent food source approach cautiously. The frogfish remains motionless, its body anchored by its limb like fins. When the distance closes to a few centimeters, the strike unfolds. Yet despite its reliance on stillness and walking, the frogfish is not incapable of rapid displacement. When threatened by a larger predator or when repositioning urgently becomes necessary, it employs an entirely different mechanism. Rather than beating fins in sustained strokes, it uses jet propulsion. On either side of its body, behind the pectoral fins, are specialized gill openings, smaller and more circular than those of many fish. By drawing water into its mouth and then forcefully expelling it through these openings, the frogfish generates a sudden burst of thrust. The body lurches forward or upward in a short, forceful movement. This jet is not sustained like the propulsion of a squid, but it is powerful enough to move the fish quickly out of immediate danger. The action is abrupt and often surprising, given the otherwise sedentary demeanor of the animal. The biomechanics of this jet propulsion rely on muscular compression of the buccal cavity. Water intake occurs through the mouth, and when the gill covers seal momentarily, pressure builds. Release through the lateral gill openings directs water backward, producing forward motion. The frogfish can angle its body to alter direction, though precision is secondary to escape in these moments. The sudden burst contrasts sharply with its usual slow gait, illustrating a dual locomotor strategy: stealth walking for hunting, explosive jetting for survival. Respiration and locomotion intertwine in this design. Gill ventilation normally serves oxygen exchange, but in the frogfish, it doubles as an emergency propulsion system. This dual use underscores an evolutionary efficiency where structures serve multiple functions depending on context. In the reef's constant interplay of predator and prey, such flexibility can determine whether an individual survives long enough to reproduce. Reproductive behavior introduces another dimension to the frogfish's ecology. Some species release eggs in buoyant ribbons that float near the surface, while others attach egg masses to substrate. Courtship often involves a brief pairing where male and female ascend together into the water column before spawning. The ascent is one of the few times a frogfish abandons the seafloor intentionally. After releasing gametes, the pair separates quickly, reducing vulnerability during a moment of conspicuous exposure. Thermoregulation in frogfish is largely passive, as they inhabit tropical and subtropical waters where temperature fluctuations are moderate. However, microhabitat selection matters. By remaining close to sponges and corals that alter local flow and shade patterns, frogfish may experience slightly buffered conditions compared to open sand. Their sedentary lifestyle conserves energy, reducing metabolic demand in environments where prey may not always be abundant. Predation risk remains real. Larger groupers, moray eels, and even octopuses may prey on frogfish if detected. Camouflage and minimal movement reduce encounters, but the frogfish's reliance on deception means that once discovered, escape options are limited. The jet propulsion burst becomes critical in such moments, offering a narrow window to relocate behind cover or wedge into crevices. As afternoon light fades into the softer tones of dusk, the reef shifts. Nocturnal predators begin to stir, and plankton density increases slightly in the water column. The frogfish remains anchored, lure flicking gently in anticipation. A small cardinal fish approaches, drawn by the simulated movement. The frogfish's body remains perfectly still, pectoral fins pressed against the substrate like hands gripping the ground. The strike, when it comes, is instantaneous. Water rushes inward, prey vanishes, and the mouth closes with silent finality. In the quiet aftermath, the frogfish adjusts its posture with a few deliberate steps, repositioning slightly to align with a new patch of sponge. The lure resumes its subtle dance. In this world of constant motion, the frogfish has chosen a different path. It walks where others swim. It bursts where others glide. It has reshaped its anatomy not for speed across distance, but for control within a confined territory. The jet-propelled walker of the reef embodies a paradox, a fish that appears immobile yet possesses explosive power, a predator that hunts through stillness rather than chase. By modifying its pectoral fins into limb-like supports and repurposing its respiratory system into a propulsion engine, the frogfish demonstrates that survival in the reef does not demand conformity to hydrodynamic ideals. Instead, it rewards specialization. In the layered complexity of coral rubble and sponge fields, where every movement is scrutinized by watchful eyes and sensitive lateral lines, The frogfish thrives by rewriting the rules of locomotion, turning the seafloor into a stage where patience and sudden force coexist in perfect balance.